Welcome to this calm documentary about the early Cretaceous and the rise of flowering plants. If you enjoy this kind of science content, please consider giving the video a like, subscribing to the channel, and sharing it with someone who might also find it relaxing and interesting. Thank you very much for spending your time here today. It really supports the channel. I'm also curious what time of day are you listening to this and where in the world are you watching from? Feel free to let me know in the comments. The early Cretaceous sits as a long interval in the history of the Mesozoic world, following the Jurassic period and preceding the later stages of the Cretaceous, and it unfolds over many tens of millions of years in which the face of the planet continues to change. During this interval, which spans roughly from the middle of the 140th million year to somewhere near the 101st million year before the present, the continents do not yet resemble the modern map, but they no longer form the single continuous landmass that existed earlier. The former supercontinent begins to separate into large northern and southern blocks, often called Laurasia and Gondwana, and the growing gaps between them fill with widening oceans and young seaways. As the crust stretches and thins, long underwater mountain chains rise along spreading ridges, and shallow basins form above slowly subsiding margins, creating broad spaces where new, marine sediments accumulate. The outline of the Atlantic Ocean starts to appear, though. In this time it is still narrow in many places, more like a series of connected interior seas than a fully developed ocean basin. And wide epicontinental seas flood onto low continental interiors wherever the global sea level rises high enough to overtop their margins. With the continents positioned closer together than in the present day, and polar regions still lacking large year-round ice sheets, the average level of the ocean stands higher, and seawater advances far inland in many regions. These flooded interiors host warm, shallow waters that support rich marine communities and lay down thick stacks of muds, sands and lime, rich deposits that later preserve fossils of plants and animals carried in from nearby land. The climate of the early Cretaceous is on the whole warmer than the climate familiar from modern times, but it is not uniform and not unchanging. Global temperatures are raised in part because concentrations of carbon dioxide gas in the atmosphere remain higher than those of the present, driven by volcanic outgassing and the slow turnover of the crust at mid-ocean ridges. Even so, the pattern of warmth varies with latitude and geography, and cooler conditions occur in some high-latitude regions, where there is evidence of seasonal snow and possibly short-lived ice in parts of the year. In many low and middle latitude belts, the air is humid and mild to hot, and precipitation patterns are shaped by the distribution of land and sea, by mountain belts that deflect air masses, and by long fetches of warm water that supply moisture to passing storm systems. There are monsoon-like circulation patterns in some regions where seasonal shifts in wind direction bring alternating phases of heavy rainfall and relative dryness, and these cycles influence river flows and lake levels and the degree of soil moisture available for plants. Fire exists as a recurring feature of many landscapes in this time, because oxygen levels in the atmosphere remain sufficient to support the spread of flames through dry vegetation, and because seasonal droughts and lightning strikes provide the necessary ignition. The likelihood and extent of fires are influenced by the mix of plant types that are present, by how tightly or loosely their communities pack biomass on the ground, and by how often dry windy conditions develop. But in this broad global perspective, all these processes are still viewed from a distance as part of a general pattern of climate and disturbance. Against this climatic background, tectonic movements continue to rearrange the distribution of continents, oceans, and mountain belts, slowly altering wind belts and ocean currents. Where continental blocks rift apart, narrow seaways open, and these channels allow warm equatorial waters to move into new areas, sometimes transporting heat toward higher latitudes, and softening temperature contrasts between equator and pole. Where plates converge, volcanic arcs rise, adding new highlands to the topography and releasing ash and gases into the atmosphere. Volcanic ash falls contribute minerals and fine particles to soils on land and sediments in lakes and shallow seas, and in some regions, repeated eruptions bury parts of the landscape under layers of fresh material that later compact into distinctive ash-rich rock units. These layers become time markers within the geological record of the early Cretaceous, helping later workers to match up events between distant regions. Life in the oceans responds to this arrangement of warmth, light, and nutrients. On the surfaces of the seas, microscopic algae and other plankton drift in the illuminated upper waters, where they harvest sunlight and form the base of marine food webs. In the deeper parts of young basins, restricted circulation and high rates of organic decay sometimes draw down oxygen in the water, leading to poorly ventilated layers in which only certain organisms can survive, and where fine sediments settle in quiet conditions to form dark, organic-rich muds. On the continental shelves and in the inland seaways, diverse invertebrate faunas populate seafloors that are often covered in sand and mud, and fish and marine reptiles patrol the water column above them. This marine world forms a continuous belt of habitat around the fragmented continents, setting boundary conditions of moisture and temperature that reach inland to the coasts and river valleys where land plants grow. On the continents themselves, the vertebrate fauna is dominated by dinosaurs and other reptiles, and early birds and mammals hold more modest roles, though the details of these communities vary from region to region. In the plant realm, non-flowering seed plants such as conifers and ginkgo relatives, together with cycads and various fern groups, form the main structural elements of many landscapes, especially at the beginning of the Early Cretaceous. The group of plants later known as flowering plants is at first either absent or present only in very restricted forms and places, and its members are not yet the dominant component of most ecosystems. Over the course of this long interval, however, the physical backdrop of warm to temperate climates elevated sea levels. Tectonic activity and shifting shorelines gradually creates and removes many kinds of habitat, offering repeated opportunities for new plant lineages to originate, spread, or decline. In the global view, the Early Cretaceous is a time of continuous but measured change in which continents slide slowly apart, seas encroach on lowlands, climate regimes vary by latitude and season, and living communities assemble and rearrange in step with the long time scales of Earth processes that shape the conditions under which early flowering plants arise and begin to diversify. During the Early Cretaceous, the arrangement of continents and oceans moves further away from the single, supercontinent state of earlier times and toward a more fragmented pattern that is still different from the modern world. In the Northern Hemisphere, the broad landmass often called Laurasia includes the areas that will later form North America, Europe, and much of Asia, but these regions are not yet separated in the ways familiar from present maps. Shallow seas extend across low-lying interiors and onto continental margins, and narrow oceanic gateways link separate basins. In the Southern Hemisphere, the large block of Gondwana holds what will become South America, Africa, Antarctica, Australia, India, and nearby fragments. Here too, rifting has begun to open new ocean basins, and long linear valleys mark places where the crust stretches and thins. In many cases, these rift zones fill with lakes and shallow marine waters that act as corridors for both marine and coastal species, and as centres of sediment accumulation that later preserve detailed records of local conditions. Along the growing Atlantic realm, the margins of the future Americas, Africa and Europe are characterised by broad passive shelves where sediment delivered by rivers spreads out onto the submerged edges of the continents. The early Atlantic is narrow in many stretches, more comparable to a chain of connected seaways and gulfs than a continuous open ocean, and the surrounding land masses shape the patterns of currents that move through it. Farther east, the Tethyan region, a wide tropical to subtropical seaway that separates Laurasia from Gondwana, forms a major pathway for warm surface waters and for the dispersal of marine organisms. To the north, parts of what will become the Arctic region are covered not by permanent ice sheets, but by shallow seas and coastal plains lying at high latitudes under long summer days and long winter nights. This global pattern of continents and seas has a strong influence on how air moves around the planet. Large continuous land areas tend to heat and cool more rapidly than oceans, and the contrast between interior continents and adjacent seaways sets up pressure differences that drive winds. In the warm seasons, interiors of continents often become relatively hot compared with surrounding seas, causing air to rise over land and drawing moist air inland from the coasts. In cooler seasons, land surfaces lose heat more quickly than water, allowing air over the continents to cool and sink, driving surface winds back toward the oceans. These seasonal reversals are not exactly the same as any modern system, because the shapes and positions of land and sea differ, but they support monsoon-like cycles in which rainfall shifts in space and time over the course of the year. Over low-latitude Tethyan waters, the combination of strong sunlight and warm surface temperatures fuels evaporation, and humid air rises to form bands of cloud and frequent storms. Where this moist air is carried on to nearby continents, some regions experience sustained wet seasons that promote dense vegetation and deep weathering of soils. In other places, the position of mountain belts and uplands blocks direct access of moist winds, leading to more seasonal or even relatively dry conditions in interior basins. Along active continental margins where one plate slides beneath another, volcanic arcs provide elevated topography that intercepts moist air masses. As air is forced to rise over these ranges, it cools and releases precipitation-feeding rivers that descend toward fallen basins and coastal plains. On the leeward sides of such ranges, air that has lost much of its moisture descends and warms, creating rain shadow effects that may limit plant growth or favour open woodlands rather than closed forests. High-latitude regions, even without large ice sheets, experience strong seasonality and light, with very long days in summer and extended periods of low light in winter. Here, continental fragments surrounded by seas are influenced by oceanic heat transport, and relatively warm currents can keep coastal climates milder than might be expected at such latitudes. This in turn allows forests composed of conifers, deciduous seed plants and ferns to extend far poleward, tolerating months of darkness and cool temperatures, but avoiding the extreme cold associated with large glacial ice. In mid-latitude settings, the presence of wide inland seas modifies regional climates by storing heat in summer and releasing it in winter, smoothing temperature contrasts and supporting relatively stable conditions on adjacent lowlands. These water bodies also host frequent fogs and mists along their margins as cool and warm air masses interact, leading to local microclimates where humidity remains high even during drier episodes. Oceans and seas not only influence air temperature and humidity but also set the path for storm tracks. Tropical cyclones and other intense storms are expected to be part of the early Cretaceous climate, though direct evidence is limited. Sedimentary features such as storm-generated sand layers, hummocky cross-stratification, and coarse lag deposits in shallow marine successions point toward episodes of strong wave action and surges. On land, heavy rainfall associated with passing storms or seasonal downpours can trigger floods on broad river plains, reshaping channels, redistributing sediment, and periodically stripping and renewing soils. These hydrological changes feed back on plant communities by altering drainage patterns, groundwater levels, and the frequency with which sites are disturbed. Over timescales of many thousands to millions of years, the gradual drift of continents changes their latitudinal positions, and with that the amount of solar energy they receive. Land masses moving toward the equator experience progressively warmer and less seasonal-like conditions, while those moving poleward face cooler and more strongly seasonal regimes. The early Cretaceous sees parts of Gondwana begin to separate with India, starting a journey that will eventually carry it through low latitudes. During these movements, coastlines are repeatedly reorganized, and new shelves, embayments, and headlands arise. Each adjustment of a shoreline changes how far moist marine air can penetrate inland, and where barriers such as coastal ranges are placed. This ongoing reconfiguration of boundaries between land and sea ensures that atmospheric circulation patterns are never entirely static over geological timescales. Although the exact values of temperature, moisture, and wind strength at specific places are not directly measured, multiple lines of geological and paleontological evidence constrain the broad picture. Fossil plant assemblages, including leaf types, and their preserved features point toward generally warm conditions in many regions, with indications of frost limited to certain high-latitude or high-elevation environments. Sedimentary structures such as evaporate deposits and calcrete horizons, when present, suggest intervals and regions of aridity or strong evaporation, while coal seams and thick organic-rich mudstones signal long-lived wet conditions and high productivity. Oxygen and carbon isotope ratios in marine fossils and sediments provide further clues about water temperatures and the global carbon cycle. Taken together, these indicators support the view that the early Cretaceous climate system operates in a greenhouse mode warmer than present on average, but with internal gradients and regions of relative coolness. Within this system, winds blow across continents that are broken into several large pieces, seas flood into interiors, and storm belts follow paths influenced by both latitude and the distribution of land and water. This dynamic but comprehensible framework sets the outer conditions under which regional landscapes, rivers, coastal lowlands and uplands evolve, and under which plant communities, including the earliest flowering plants, will occupy, adjust to, and reshape the environments available to them. During the early Cretaceous, the broad arrangement of continents, seas, and atmospheric circulation patterns translates on the ground into a set of recognizable regional terrains. where land plants can root, grow and spread. Along many continental margins, especially around the young Atlantic realm, and the Tethyan seaway-wide low-lying coastal plains grade gently into shallow marine shelves. These coastal zones are built from layers of riverborne sand and mud that prograde step by step into the sea, interrupted at times by shore-face sands reworked by waves and storms. Farther inland from some of these coasts lie expansive interior basins, areas of the crust that slowly subside under their own weight, or under the load of adjacent mountain belts. In these basins, rivers, lakes and wetlands occupy the lowest parts of the topography, and surrounding slopes send sediment and dissolved minerals downslope to fill depressions and nourish developing soils. Where continents meet convergent plate margins, in contrast, long chains of mountain ranges rise flanked on their inland sides by foreland basins, and on their seaward sides by narrow coastal strips. Together, these elements of coastal plains, shelves, interior basins and uplands create a patchwork of settings, each with its own typical water table depth, soil development and disturbance regime, even before considering the specific plant communities that occupy them. On many low-latitude and middle-latitude margins, coastal plains stretch tens to hundreds of kilometers inland from the shoreline. They are crossed by branching networks of rivers that carry water and sediment from uplands toward the sea, shifting position over time as channels migrate, laterally split or abandon old paths. Between the channels, flat to gently undulating interflows stand slightly higher above the regional water table. During wet periods, floodwaters spill out of the channels, spread across nearby floodplains, and lay down thin layers of silt and clay, slowly building natural levees along the banks and freshening the surface soils. During drier intervals, the same floodplains may remain aerated for longer periods, allowing roots to penetrate deeper, and soil horizons rich in organic matter or iron compounds to form. Near the seaward edge of these plains, the zone where river influence meets tidal and wave processes typically contains tidal flats, marshes and coastal lagoons, though the exact mix varies from place to place. Here, frequent shifts between submergence and exposure, along with the mixing of fresh and saline waters, impose strong filters on the organisms that can live there. The shallow marine shelves beyond, rarely more than a few tens to perhaps a few hundred meters deep during this time, receive a rain of sediment and organic particles from above and from the land, and in calm intervals support benthic communities that in turn become entombed in accumulating strata. Interior basins, some tied to earlier rifting events, and others to flexure near mountain belts, form another prominent class of early Cretaceous landscapes. Many of these basins have low overall relief with broad, gently sloping surfaces, drained by meandering rivers, and local depressions occupied by lakes of varying size and longevity. The balance between input of water from rivers and rainfall on the one hand, and loss of water through evaporation and outflow on the other, controls whether a basin floor hosts permanent lakes, seasonally fluctuating water bodies, or ephemeral player-like flats. In more humid sectors, long-lived lakes can develop stratified water columns and thick successions of fine-grained sediments on their floors, preserving detailed seasonal or annual layering, along with pollen spores and fragments of leaves that settle into them. In more arid or strongly seasonal sectors, lake levels may rise and fall, repeatedly generating shoreline features at different elevations and concentrating dissolved salts whenever evaporation outpaces inflow. Around the edges of these basins, Piedmont zones receive sediment from nearby uplands in the form of alluvial fans and sheet floods, creating coarse, rapidly aggrading surfaces where channels are unstable and soils are repeatedly buried and reformed. On slightly more stable slopes, finer sediments and volcanic ash can accumulate, and weather giving rise to thicker soil profiles that are better able to support sustained plant growth. Upland regions, including both modest highlands and more pronounced mountain belts, contribute another important element to the early Cretaceous terrain. In tectonically active areas, steep slopes are cut by rivers that incise into bedrock and transport coarse gravel and boulders downslope during floods, while between these valleys, ridges and plateaus rise to elevations where temperatures are cooler and conditions more seasonal than on adjacent lowlands. Weathering on these upland surfaces generates regolith and soils that vary in thickness and fertility, depending on rock type, climate and time. In some cases, resistant rocks maintain thin, stony soils with relatively sparse plant cover, while in others, mafic or volcanic parent materials give rise to more nutrient-rich substrates that can support denser vegetation where moisture is adequate. Snow may fall seasonally on the highest peaks at suitable latitudes, feeding meltwater into headwater streams and influencing the timing of runoff. Over long intervals, glacial ice is not thought to be widespread, but localized cold conditions are still possible at high elevations or latitudes, and these areas would mark the cooler end of the spectrum of terrestrial environments in which plants could live. Between the extremes of low coastal plains and higher mountain belts lie varied plateaus, dissected uplands, and gently rolling surfaces that form much of the land area of some early Cretaceous continents. Here river systems cut valleys of moderate relief with interfluves, supporting soils of intermediate depth and drainage. In many such regions, volcanic activity supplies periodic inputs of ash, which settle out over wide areas and add easily weathered glass and mineral grains to the surface. Over time, these materials break down to yield clay-rich horizons and release nutrients such as phosphorus, potassium, and calcium, which become available to plants and to microbes in the soil. The interplay of volcanic inputs, chemical weathering, and biological activity slowly transforms initial mineral substrates into complex soil profiles, with distinct layers ranging from organic rich tops to leached middle zones and accumulations of secondary minerals below. In warm, humid climates, intense weathering can strip many of the more soluble elements from the upper parts of the profile, concentrating iron and aluminium oxides, and giving soils a characteristic reddish or yellowish colour. In cooler or drier settings, weathering proceeds more slowly, and soils retain more of the original mineral signatures of their parent rocks. Across all these terrains, from coastal plains and interior basins to uplands and plateaus, water movement through the ground and along the surface exerts strong control on where plants can establish and how they grow. The position of the water table relative to the land surface, the frequency and depth of flooding, the rate of drainage after heavy rainfall, and the degree to which soils dry out between wet periods all govern the availability of oxygen to roots, the mobility of nutrients, and the likelihood of erosion or burial. Low-lying floodplains that sit close to the water table may maintain moist or waterlogged conditions for extended periods, favouring plants tolerant of anoxic or saturated soils, while slightly higher levees and terraces may offer better drained settings where deeper root systems can develop. On sloping ground, thin soils may be prone to downslope creep and landsliding, disturbing plant cover at intervals and creating a mosaic of stable and recently disturbed patches. In contrast, broad interfluves on gently rolling surfaces may experience relatively infrequent physical disturbance, but gradual changes in soil chemistry and structure through time. These linked regional elements of relief, substrate hydrology, and disturbance define the physical template on which early Cretaceous vegetation patterns are built. Without yet specifying which groups occupy which sites, it is clear that the planet offers a wide range of potential niches, from waterlogged delta plains and lake margins to well-drained river terraces and rocky upland slopes. As continents continue to shift and as climate patterns evolve within the greenhouse framework, the relative extent and configuration of these terrains change, opening new areas to colonization, isolating others, and continually reshaping the three-dimensional canvas of land surfaces on which plant lineages, including the emerging flowering plants, will establish their roots and expand their ranges in the landscapes of the early Cretaceous world. Across the early Cretaceous continents, the regional terrains of coast basins and uplands are rarely static. They are reshaped again and again by moving water, shifting sediment, fire, volcanic activity, and smaller physical disturbances that together produce a landscape that is patchy in space and time. On broad coastal plains, rivers migrate laterally across their floodplains, eroding material from one bank and laying it down on the other, so that channels swing in wide bends and sometimes cut off loops to leave behind oxbow lakes and abandoned reaches. Each avulsion where a river suddenly shifts to a new course leaves a former channel belt high and relatively dry compared with the new course, and resets the local pattern of water tables and soil moisture. In some intervals, sediment supply from uplands is high, and rivers build their floodplains vertically, through repeated flood deposits raising natural levees, and periodically overtopping them to spread thin sheets of silt and sand over interfluves. At other times, when discharge increases beyond the capacity of channels, to contain it, large floods strip away parts of the floodplain, exposing older layers and creating open surfaces of fresh sediment. These events can bury low-lying ground, smothering existing vegetation, and open new bare patches that gradually stabilize as roots and microorganisms colonize them. In deltas where rivers meet standing bodies of water such as lakes, lagoons, or inland seas, the interplay between river flow wave action and tides constructs intricate networks of distributary channels, sandbars, and interdistributary bays. Here the position and shape of land surfaces can change on timescales of years to centuries as channels bifurcate, choke, and reoccupy former paths. Some lobes prograde seaward until they become unstable and are abandoned while new lobes begin to grow elsewhere along the shoreline. This switching behaviour leaves behind a patchwork of sub-environments, including levees, crevasse splays, shallow ponds, and muddy flats that alternate between submergence and exposure. Sediment compaction and subsidence combined with ongoing delivery of new material cause parts of the delta plain to sink and others to build upward altering local drainage patterns and the distribution of permanently versus seasonally waterlogged ground. In interior basins and on Piedmont surfaces near mountain fronts, alluvial fans contribute to the disturbance mosaic. These fan-shaped accumulations of gravel, sand and mud spread out from canyon mouths, built by short-lived but powerful flows that occur when intense rainfall or sudden snowmelt mobilizes debris. During quiet intervals, individual channels carry water and sediment along the fan surface, but during major events they can rapidly shift position, carving new paths and abandoning old ones. The result is a surface that is reworked at irregular intervals, with some patches remaining stable for long periods, and others experiencing frequent burial or erosion. Such heterogeneity in disturbance history leads to differences in soil development across short distances, with older, less disturbed patches supporting thicker, more chemically altered profiles than younger, recently reworked ones. Volcanic activity introduces another layer of episodic change. Along active margins and over volcanic provinces, eruptions eject ash lapilli and larger fragments into the atmosphere which then settle out over surrounding regions. Fine ash can travel hundreds of kilometers downwind, dusting wide areas with a blanket of fresh, glassy particles. In some cases, the thickness is small, and the main effect is to add a thin layer of easily weathered material to existing soils. In other cases, especially closer to vents, thick accumulations of ash and pyroclastic flow deposits can bury entire landscapes. These deposits may be meters to tens of meters thick, abruptly cutting off existing root systems and smothering surface communities. Over time, weathering converts volcanic material into clay-rich horizons and releases nutrients that can support vigorous plant growth. But in the short term, the sudden change in surface conditions represents a major disturbance. Lava flows, though less extensive in many vegetated lowlands, also create sharp boundaries between fresh bare rock and surrounding older surfaces, adding to the fine-scale variability in substrate age and character. Fire operates within this tectonic and sedimentary context as a recurring surface disturbance. In regions where climate provides a dry season and fuels accumulate lightning, or in some cases volcanic activity, can ignite fires that spread through grass like understory shrub layers and forest litter. The frequency and intensity of these fires depend on the balance between fuel production, fuel moisture, and ignition sources. In some humid tropical belts, fires may be relatively infrequent and localized, while in more seasonal regions they may occur at intervals short enough to influence the long-term composition and structure of vegetation. Fire consumes above ground, biomass volatilizes some nutrients and converts others into ash that can be leached into soils or washed downslope by rain. Post-fire surfaces are exposed to increased sunlight at ground level until new foliage develops, and the combination of charred material and bare patches can alter surface runoff and erosion rates during subsequent storms. Repeated fires through time create a mosaic of patches at different stages since the last burn, each with its own structure of vegetation and soil cover. Storm systems and associated wind events contribute further to the disturbance regime, especially in coastal and low-latitude regions. Strong storms raise water levels along shorelines, generating waves and currents that erode beaches, barrier islands and coastal dunes, and transport sand landward or along the coast. Storm surges can push seawater into river mouths and across low coastal plains, depositing sheets of sand and shell material over previously muddy or peaty surfaces. Within forests and woodlands, high winds can topple individual trees or groups of trees, opening gaps in the canopy and exposing the forest floor to increased light and to physical damage from falling trunks and branches. These windthrow events create small patches where soils are disturbed by uprooted root masses and where coarse woody debris accumulates. Over longer periods, changes in relative sea level, driven by a combination of global processes and local subsidence or uplift force coastlines, to migrate landward or seaward. During phases of relative sea level rise, transgression floods coastal plains and shifts environments of deposition inland, while during phases of relative sea level fall, regression allows rivers to incise and extend their systems seaward. These shifts rearrange the positions of estuaries, deltas, and nearshore environments, effectively moving disturbance regimes such as tidal flooding, storm wave attack, and delta lobe switching across the landscape. In parallel, climatic fluctuations within the overall greenhouse framework can modestly alter precipitation intensity, storm tracks, and seasonality, which in turn change flood frequency, fire regimes, and the balance between erosion and deposition. At smaller scales, processes such as soil creep, frost heave in cooler regions, animal burrowing, and the growth and decay of roots continually rework the uppermost layers of the ground. Though individually subtle, these processes summed over large areas and long periods contribute to the constant renewal and adjustment of soil structures, pore networks, and microtopography. They interact with the larger, more dramatic disturbances by influencing how water infiltrates, how easily slopes fail, and how quickly new substrates can be colonized by organisms once a major event has passed. Taken together, these varied disturbances ensure that early Cretaceous landscapes are not uniform carpets of stable ground, but are instead composed of mosaics of patches differing in age, substrate drainage, and exposure. Some patches are freshly created by floods, ash falls, lava flows, storms, or fires. Others are intermediate, having experienced disturbance in the recent past, and now undergoing soil formation and gradual stabilization. Still others are comparatively old, with well-developed soils and long-established surface forms. Through time, patches shift from one state to another as new disturbances occur and old ones fade into the background. This dynamic patchwork sets the stage on which plant communities assemble in different ways in different places depending on their tolerances for burial drought, waterlogging, and heat, and on their ability to exploit the opportunities created by the continual reshaping of the early Cretaceous land surface. Before flowering plants become common features of terrestrial ecosystems, the regional terrains of the early Cretaceous are covered and structured by plant groups that have already dominated landscapes for many millions of years. On many low-latitude coastal plains, broad river valleys, and humid interior basins, tall conifer forests form the main vertical framework of vegetation. These conifers include forms with needle-like foliage, and others with flatter scale-like leaves, and they often carry their reproductive cones high in the crown. Trunks rise straight from the ground, closely spaced in some stands, and more widely separated in others, and the canopy intercepts much of the incoming light. Beneath this canopy, a shaded understory develops in which ferns, small seed plants, and various shrubby forms occupy the limited light that filters down. In places where the canopy is dense, the forest floor may be covered by a continuous layer of litter, composed of needles, twigs, cones, and fragments of branches intermingled with fronds and stems from lower plants. This litter layer moderates soil temperature and moisture, slows the movement of water across the surface, and provides organic matter that decomposes at different rates depending on the local climate and the composition of the material. In more open stands, where conifer crowns do not fully close patches of sunlight reach the ground, and support denser fern thickets, early shrubs and low-growing seed plants adapted to partial shade. On some parts of these plains, cycads are prominent elements of the vegetation. Cycads are seed plants with stout, often unbranched trunks and crowns of tough pinnate leaves that radiate from the top of the stem. They can form scattered individuals within conifer forests or gather into small groves on better drained slopes and terraces. Their robust leaf bases and persistent leaf crowns create distinct microhabitats around their bases, where litter accumulates and roots explore the upper layers of soil. The presence of cycads is often associated with well-drained to moderately drained soils, and their distribution reflects the balance between moisture availability and the risk of extended waterlogging. In some regions, especially on nutrient-rich substrates, or on older, more stable surfaces, ginkgophytes contribute to the canopy or sub-canopy. These seed plants bear fan-shaped leaves with a characteristic venation pattern and can adopt tree or large shrub forms. Their leaves shed seasonally in some climates add to the annual input of organic material to the soil and may indicate environments with marked seasonal changes in light or temperature. Together, conifers, cycads, and ginkgophytes create a range of forest types from relatively closed evergreen stands in humid zones to more open, mixed assemblages where seasonal dryness or poorer soils limit canopy density. Ferns are abundant across many of these landscapes not only as understory components of forests, but also as dominant plants in some open or disturbed habitats. In waterlogged floodplains along the edges of lakes and swamps, and on recently deposited sediments, where seed plants have not yet fully established large tree ferns, with trunk-like stems can form low canopies, while smaller ferns fill in beneath them. Their fronds intercept light close to the ground and create dense layers of vegetation that stabilize sediments and trap fine particles. The spores they produce, carried by air and water, allow rapid colonization of new surfaces exposed by floods, volcanic ash falls, or fires. Smaller ferns, including those with creeping rhizomes, can quickly cover bare ground, reducing erosion, and beginning the cycle of organic accumulation that leads to soil development. Along stream margins and in low-lying depressions, horsetails and other spore-bearing plants related to ancient club mosses often form dense stands. Their jointed stems and whorled branches rise in uniform clusters, rooted in saturated or periodically inundated soils. These plants tolerate episodes of flooding and burial by fine sediments and can re-establish quickly after disturbance, making them typical pioneers in zones that experience frequent changes in water level or sediment load. On slightly higher ground and better drained substrates, low shrubs and small non-flowering seed plants occupy gaps between larger trees. Some of these are seed ferns, a group that combines fern-like foliage with seeds borne on specialized structures, and they may persist locally in the early Cretaceous in certain regions. These plants add to the diversity of forms and strategies in the understory and midstory, each exploiting particular combinations of light moisture and soil chemistry. Moving away from the wettest lowlands, the structure of plant communities changes. On well-drained river terraces, older levees and parts of interior uplands, forests may open into woodlands where trees are more widely spaced, allowing grasses in the modern sense to be largely absent, but leaving room for herbaceous plants, shrubs, and low fern carpets. In these more open settings, light reaches the ground more consistently, and plants adapted to higher irradiance and greater temperature range can thrive. Soil profiles here tend to be better aerated with clearer differentiation into organic rich surface horizons and more mineral-rich layers below. Root systems of trees and shrubs can penetrate deeper into the profile, accessing stored moisture and nutrients. And the overall plant community reflects a balance between water availability, soil depth, and disturbance frequency. On upland slopes and in cooler or more seasonal climates, conifers often dominate, forming forests in which evergreen foliage is adapted to withstand periods of relative dryness, frost, or low nutrient availability. Needle-like leaves with reduced surface area and thick cuticles help limit water loss and allow trees to retain foliage for several years, which in turn influences nutrient cycling as needles fall and decay slowly. In some high-latitude regions that experience long periods of darkness in winter and extended daylight in summer, evidence from fossil wood and leaf deposits suggests that some seed plants adopt a deciduous habit, shedding leaves or modifying growth patterns to cope with seasonal extremes. Under these canopies, ground layers continue to be built from ferns, mosses, and small shrubs, but their composition differs from that of low-latitude forests, reflecting the cooler and more strongly seasonal conditions. Wetlands, swamps, and peat-forming environments are also common components of early Cretaceous landscapes. In poorly drained depressions along the margins of lakes or shallow seas, and in parts of coastal plains where the water table sits close to or above the surface for much of the year, plant communities dominated by water. Tolerant conifers, tree ferns, and other moisture-loving plants accumulate large amounts of organic matter. Because decomposition is slowed under waterlogged low-oxygen conditions, peat can build up, eventually forming thick accumulations that, when buried and compacted, become coal. These peatlands act as long-term reservoirs of carbon and influence local hydrology by storing and slowly releasing water. The floras of such wetlands are typically distinct from those of adjacent well-drained areas, with a higher proportion of species adapted to saturating conditions and to the mechanical stress associated with fluctuating water levels. Across all these habitats, from saturated swamps to upland woodlands, non-vascular plants such as mosses and liverworts cover many surfaces. They colonize rocks, fallen logs, and soil patches, holding moisture close to the surface and contributing fine organic material as they grow and decay. Their presence helps to stabilize substrates and provides microsites for the germination and establishment of larger plants. Lichens associations between fungi and photosynthetic partners are likely present as well, especially on exposed rocks and tree trunks, though their direct fossil record is sparse. Together, these communities of vascular and non-vascular plants carry out high rates of photosynthesis, fix carbon, and drive the cycling of nutrients such as nitrogen and phosphorus through uptake, litterfall, and decomposition. The structure of the pre-flowering plant world therefore varies strongly across the early Cretaceous terrain. In some regions, continuous canopies of conifers or mixed seed plants form dense forests. In others, mosaics of woodland, shrubland, fern, dominated clearings and open wetlands appear often structured by underlying gradients in water. Table depth, soil age and disturbance history. Coastal plains close to the sea may host mixed forests transitioning into marshes and tidal flats, while interior basins may hold extensive swamp forests around lake margins and more open vegetation on drier divides. Uplands contribute yet another set of communities, often with lower stature vegetation, more exposure to wind and temperature extremes and soils that differ in chemistry and depth from their lowland counterparts. Within this array of settings, the groups that dominated earlier periods continue to shape the physical and biological environment. They regulate the amount of light that reaches the ground, influence how water moves through soils, and returns to the atmosphere and create the litter roots and dead wood that support diverse microbial and invertebrate communities. Their distribution sets the baseline pattern of habitats into which new plant lineages will eventually integrate. At this stage of the early Cretaceous narrative, the world is still largely structured by these long-established lineages, and their forests, woodlands and wetlands define the main forms of terrestrial vegetation that blanket the dynamic landscapes shaped by rivers, basins, volcanoes, and climate. In the midst of the familiar conifer forests, fern thickets, and cycad groves of the early Cretaceous, a new kind of seed plant begins to appear in the geological record. These are the flowering plants or angiosperms, and at the scale of the planet their first traces are modest and scattered rather than sudden and overwhelming. In some basins and along particular river systems, small fragments of leaves, tiny reproductive structures and distinctive grains of pollen indicate the presence of lineages that bear their seeds enclosed within protective tissues rather than exposed on the surfaces of cones. The earliest evidence of such plants comes from fine-grained sediments laid down in lakes, quiet river channels, and low-energy floodplains, often where preservation of delicate material is especially good. In these settings, leaf impressions, compressions of stems with attached organs, and accumulations of pollen and spores settle out of the water column or fall directly onto soft muds and silts, later to be buried by further layers of sediment. In some lacustrine deposits of Western Eurasia and Eastern Asia, for example, assemblages of small aquatic or semi-aquatic plants appear in rocks of early Cretaceous age. Their fossils show finely divided leaves and reproductive organs that suggest a flowering plant affinity, even though their exact relationships within the group remain debated. In other regions, such as parts of what is now North America and South America, isolated leaves and pollen grains with features characteristic of flowering plants occur in fluvial and deltaic sequences that record the shifting courses of rivers and the slow building of coastal plains. These finds, though often incomplete, mark the gradual entrance of angiosperms into a world already structured by older plant groups. The habitats in which early flowering plants are most frequently recorded share several broad characteristics. Many are associated with freshwater environments such as lakes, ponds, and the quieter reaches of river systems where water levels are relatively stable over at least part of the year and where fine sediments accumulate. In these places, early angiosperms may grow rooted in shallow water, on exposed mudflats, or on low riparian banks that are periodically flooded but also experience intervals of exposure. The structure of their remains suggests that some forms are herbaceous with thin stems and small leaves that complete their life cycles quickly, while others have more robust, possibly shrubby habits. Some fossils show elongated strap-like leaves, others display broad laminae with clear networks of veins. These venation patterns, together with the arrangement of attachment points to stems and the presence of enclosed seeds or fruits in associated material, support their placement within the flowering plant lineage. However, the full range of life forms present at this early stage is not entirely known because preservation favours certain plant parts and habitats over others. In more disturbed settings such as active floodplains and the margins of deltas, evidence suggests that early angiosperms occupy pioneer roles in patches of ground opened by erosion deposition or local treefall. Thin silt and sand layers that cap older surfaces sometimes contain abundant small leaf fragments and seeds of flowering plants mixed with debris from ferns, horsetails and gymnosperms. This pattern implies that in some regions, flowering plants are able to establish quickly on fresh substrates, perhaps benefiting from shorter generation times and flexible growth strategies. The mosaic of channels, levees, crevasses, splays, and abandoned channels that characterize early Cretaceous floodplains offers numerous small sites where competition from established woody vegetation is temporarily reduced. Early angiosperms with relatively small stature and possibly rapid reproductive cycles can exploit these microhabitats between more permanent stands of conifers and other seed plants. Coastal environments also provide opportunities for these new plants. Along the landward margins of lagoons and estuaries where freshwater and seawater mix, and where sediments shift under the influence of tides and storms, communities of salt-tolerant and disturbance-tolerant plants form. Although the direct fossil record of flowering plants in these marginal marine settings is less continuous than in some freshwater basins, occasional finds of leaves and reproductive structures in shallow marine sediments suggest that at least some angiosperms reach brackish or nearshore environments during the early Cretaceous. Their remains sometimes occur alongside those of conifer driftwood fern fronds and spores of wetland plants, indicating that plant material from multiple habitats is washed together and buried. Sorting out which fragments represent local coastal vegetation and which are transported from inland sites requires careful sedimentological and paleobotanical analysis, and in many cases uncertainty remains. The global distribution of early flowering plant fossils is patchy, reflecting both the original ecology of these plants and the incomplete nature of the geological record. Some regions with extensive early Cretaceous sedimentary successions show abundant evidence of angiosperms in certain intervals and settings, but little in others, suggesting that their presence is influenced by local climate substrate and disturbance patterns. Other regions preserve long records dominated by non-flowering seed plants and ferns, with only rare or late-appearing angiosperm remains. It is therefore likely that early in their history flowering plants are not evenly spread across all continents and climates, but instead occupy particular environmental niches, where their traits provide advantages while remaining absent or rare in others. As time passes within the early Cretaceous, more diverse assemblages of angiosperm leaves and reproductive structures appear in some basins, indicating a gradual expansion in both taxonomic diversity and ecological breadth. The earliest known flowers preserved with enough detail to study show relatively simple arrangements of organs, often lacking the complex layering and specialization seen in many modern forms. Their petals or petal-like structures when present are modest in size, and their reproductive parts are exposed enough that direct contact with water, wind or animals could transfer pollen. Some fossils show seeds enclosed in small, often elongate or rounded structures that can reasonably be interpreted as early fruits. The variety of these forms through successive layers hints at evolutionary experimentation with different architectures and reproductive strategies. Still, it is important to note that many aspects of these early flowers, including their original colours, scents, and some aspects of their three-dimensional organisation, are not recoverable from the available material. In sediment cores and rock sections where palynologists examine microscopic remains, the appearance of pollen grains with distinctive angiosperm features provides an additional line of evidence for the early spread of flowering plants. These grains often have complex wall structures and apertures that differ from those of contemporary gymnosperms, and their first appearance in a given region can precede or accompany the arrival of clearly identifiable angiosperm leaves and flowers in the macrofossil record. Because pollen can be transported by wind and water over considerable distances, its distribution must be interpreted with care, but when combined with other fossil evidence and with knowledge of the depositional environments, it allows scientists to trace broad patterns of angiosperm presence and diversification across space and time. All of this evidence taken together depicts a world in which flowering plants begin the early Cretaceous, as minor, often local components of vegetation occupying specific niches along water margins on frequently disturbed ground and in some sheltered aquatic environments. They are not yet the dominant trees of vast forests, nor are they present in all climatic zones. Instead, they form part of a complex mosaic of plant life interwoven with conifers, cycads, ginkgophytes, ferns and other groups. Their enclosed seeds and varied leaf forms suggest capacities for flexible reproduction and growth, but the full extent of these capacities and their precise ecological roles remain topics of ongoing investigation. Within the bounds of what the fossils clearly show, the emergence of angiosperms in the early Cretaceous can be described as a gradual, spatially uneven process in which new plant lineages appear first in particular habitats and basins, recorded as scattered leaves, delicate reproductive parts and microscopic grains, against the backdrop of long-established plant worlds that still define most of the structure and function of terrestrial ecosystems. As the early Cretaceous progresses, some forests that were once dominated almost entirely by conifers, cycads and other long-established seed plants begin to show a more complex mixture of canopy and understory elements. In these places, flowering plants move beyond the most marginal and disturbed settings and become regular, though still not exclusive components of forest mosaics. On broad river plains and low coastal terraces, patches of woodland that once consisted mainly of tall conifers with relatively simple vertical structure start to include angiosperm shrubs and small trees in the lower and middle levels of the vegetation. These new elements occupy spaces beneath and between conifer crowns along forest edges and in gaps created by windthrow fire or channel migration. The overall effect is not a uniform replacement of older plant groups, but a diversification of forest structure with more pronounced layering and local variation. In some regions, conifers continue to supply the tallest emergent trees, forming an upper canopy that controls much of the interception of light and rainfall. Below this, an intermediate tier of angiosperm shrubs and young trees can develop, where light levels are sufficient. Their leaves, often broader and thinner than those of neighbouring gymnosperms, capture diffuse light under the canopy and contribute to a denser, more continuous foliage layer. In small gaps where canopy trees have fallen, angiosperm saplings can grow more rapidly, at least in some cases, than the seedlings of slower-growing conifers, allowing them to fill openings quickly and form temporary or semi-permanent patches dominated by flowering plants. In other gaps, gymnosperms regain control, and the local dominance of angiosperms waxes and wanes in step with disturbance and competition. Evidence for these mixed forests comes from fossil leaf beds and wood assemblages in fluvial, deltic and coastal plain deposits. In some early Cretaceous successions, layers of fine sandstone and siltstone preserve leaf mats that combine broad-leaved angiosperm foliage with conifer needles, cycad leaflets, and ginkgophyte leaves. The relative proportions of these elements vary between beds that were laid down only a short time apart, suggesting that different patches of forest with different compositions contributed plant material to channels at different times. On nearby floodplain surfaces that escaped major erosion, buried soils show traces of roots at several depths and horizons, indicating that multiple plant types with different rooting strategies coexisted. In some cases, fossil wood recovered from such strata shows vessel-bearing tissues characteristic of angiosperms alongside trachid-based conifer wood, pointing towards stands in which both groups were present at the same sites. Not all early Cretaceous regions show this level of mixing. Some high-latitude and upland forests remain dominated by conifers and other gymnosperms with little or no evidence of flowering plants, at least within the intervals captured by the rock record. In low-latitude basins with particularly wet or nutrient-poor conditions, swamp forests may continue to be formed mainly by water-tolerant conifers, tree ferns and other non-angiosperm groups. However, in more seasonally wet and well-drained settings, particularly along active river systems, and on the margins of lakes and deltas, mixed forests are increasingly documented. In these environments, the presence of flowering plants modifies the distribution of light within stands. Broad-leaved angiosperms often have canopy architectures that intercept light differently from those of conifers, creating patches of deeper shade under their crowns and patches of higher light in the spaces between them. Their leaves may be held more horizontally and in denser clusters so that small changes in their abundance can alter the pattern of sunflecks and shade on the forest floor. This in turn affects the growth of understory ferns, horsetails and small shrubs that depend on particular light regimes. Leaf-fall patterns also change as angiosperms become more common in forest mosaics. Many early flowering plants are thought to produce leaves with shorter individual lifespans than those of some evergreen gymnosperms, though the evidence is not uniform across all taxa. Where leaves are shed annually, or on relatively short timescales, the litter layer receives frequent inputs of thin nutrient-rich material that decomposes more quickly than thicker, tougher conifer needles or cicad leaves. Decomposition rates are influenced by local climate and soil biota, but in general more labile angiosperm litter can support higher microbial activity and faster nutrient turnover, at least in patches where it accumulates. This does not mean that all angiosperm litter decomposes quickly, or that all gymnosperm litter decomposes slowly, but the introduction of a new range of leaf types increases the variability of litter quality on the forest floor. In places where this litter accumulates, soil structure and chemistry begin to reflect the combined influence of different plant groups. Thin, broad angiosperm leaves fragment into small pieces that mix with fine mineral grains and organic matter from other sources, contributing to the formation of aggregated soil structures that retain moisture and air in small pores. The presence of these aggregates can enhance root penetration and water infiltration, especially in upper soil horizons. Meanwhile, persistent conifer needles and woody debris create larger pieces of slowly decaying material that form a coarse framework within the litter layer. The result is a heterogeneous surface horizon with microhabitats that differ in moisture aeration and nutrient availability over centimetre to metre scales. Root systems of angiosperms and gymnosperms interact within this mixed substrate. Some flowering plants develop relatively fine, densely branched root networks near the surface, exploiting shallow resources and responding quickly to variations in moisture and nutrient supply. Conifers and other deep-rooted gymnosperms may extend roots further into the profile, tapping stored water and nutrients in deeper layers. In combination, these rooting patterns can lead to more complete occupation of the soil volume, by roots potentially reducing losses of nutrients to leaching and affecting how water moves through the profile. Again, the exact balance between these effects varies between sites, and the fossil record does not allow detailed reconstruction of root architectures everywhere. However, fossil soils with mixed root traces of varying diameters and orientations support the idea of complex root systems in mixed forests. In terms of vertical structure, some early Cretaceous forests likely exhibit 3 or more recognizable strata: an upper canopy, an intermediate layer of small trees and tall shrubs, and a lower understory of herbs and ferns. Angiosperms participate in the intermediate and lower strata from relatively early in their history, while gymnosperms and older seed plants continue to dominate much of the upper canopy in many regions. Over time, there are indications that some angiosperms also reach larger sizes, contributing more directly to the canopy, but within the early Cretaceous, these developments appear to be in progress rather than complete. In some fossil assemblages, larger diameter angiosperm wood with ring-like vessel arrangements suggests the presence of substantial shrubs or small trees, though well-preserved examples are not abundant. The spatial arrangement of these mixed forests is also heterogeneous. Along major river corridors, belts of riparian vegetation follow channels across the landscape, with compositions that can differ from those of more distant floodplain or terrace forests. Early angiosperms are often more strongly represented in these riparian belts, where frequent disturbance and higher nutrient availability offer opportunities for rapid establishment. Farther from channels on older, more stable surfaces, conifer-dominated stands may persist with only sparse angiosperm representation. On gently sloping interfluves woodland with a relatively open canopy may support a higher proportion of shrubs and low trees, including angiosperms, especially in climates with pronounced seasonality or in soils influenced by volcanic ash. Where volcanic inputs are significant pulses of nutrient-rich material from ashfalls can temporarily enhance soil fertility, supporting flushes of plant growth in which angiosperms may compete effectively. In coastal plain settings, the transition from inland forests to nearshore wetlands and tidal environments passes through zones where mixed stands of conifers, angiosperm shrubs and ferns occupy slightly elevated levees and back swamps. Their differences in tolerance to salinity and waterlogging determine which species dominate particular microhabitats. Some early angiosperms appear in association with brackish conditions, but detailed reconstructions of their tolerance ranges remain uncertain. What is clearer is that the overall pattern is not one of uniform vegetation bands, but of interlocking patches of differing composition and structure, arranged along subtle gradients in elevation, soil properties, and disturbance history. The presence of angiosperms in these mosaics adds complexity to the way plant communities respond to and recover from disturbance. After a floodfire or ashfall, pioneer angiosperms can sprout or germinate quickly in open areas, forming low-dense stands that protect the soil and alter light availability for subsequent colonists. Gymnosperms and other slower establishing plants gradually re-enter these patches, and over time some sites return to conifer-dominated states while others retain a significant angiosperm component. This shifting balance over successive disturbance cycles likely varies between regions and climates, and the fossil record with its time-average deposits offers only snapshots of these dynamics. Still, the repeated association of angiosperm remains with sediments that represent younger, more disturbed surfaces, and their co-occurrence with gymnosperm material in more mature floodplain and terrace deposits supports the view that angiosperms participate in multiple stages of early Cretaceous forest succession. Within the limits of available evidence, the mid to late early Cretaceous can thus be described as a time when many terrestrial landscapes host forests and woodlands built from a mixture of old and new plant groups. Conifers, cycads, ginkgophytes, ferns and other established lineages continue to form the backbone of vegetation, especially in certain climates and geomorphic settings. Flowering plants, though still limited in overall diversity compared with later times, become increasingly embedded within these communities, shaping and being shaped by local conditions of light soil and disturbance. The forests that result are more varied in canopy structure, leaf form, litter composition and root organization than those of earlier periods, and this added heterogeneity at the level of habitats and communities sets the context for further changes in specific environments such as wetlands, lake shores and lagoon margins where early angiosperms also play growing roles. In the early Cretaceous, many of the most important places where flowering plants appear and spread lie in and around wetlands, lakes and sheltered lagoon margins. These environments occupy low points in the landscape, where water collects and persists either as standing bodies or as saturated ground, and they host plant communities that differ in clear ways from those of adjacent well-drained terraces and slopes. In broad interior basins, lakes of various sizes fill depressions created by subsidence faulting or past erosion. Some of these lakes are long-lived with stable shorelines and layered accumulations of mud and silt on their floors. Others are more changeable, expanding and contracting as climate shifts, as river inflow varies, or as outlets are opened or blocked. Around their margins, the combination of shallow water, fine sediment and fluctuating water levels creates gentle gradients from fully aquatic zones to permanently wet soils and then to only seasonally saturated ground. Along these gradients, belts of vegetation organize according to differing tolerances for water depth, waterlogging and disturbance. In the shallowest parts of lakes, rooted aquatic plants grow partially or completely submerged, anchoring themselves in soft sediments and extending leaves and stems into the water column. The fossil record captures some of these as delicate impressions of narrow leaves whirled around thin stems, preserved in fine-grained lake muds. In slightly deeper water, plant fragments may be transported and mixed with planktonic remains, making it harder to distinguish local growth from drifting material. Closer to shore in water that is often less than a few tens of centimeters deep, emergent vegetation takes hold. Here, stems and leaves rise above the water surface while roots and lower stems remain submerged. These zones, which are regularly influenced by waves generated by storms and by local wind support, sturdy plants able to withstand gentle movement of water and occasional burial by suspended sediment. Tree ferns rush like plants, and early angiosperms with herbaceous or low shrubby forms are all present in different combinations depending on the specific lake and climate. On the landward side of the emergent zone, where soils are saturated but rarely flooded deeply, peat-forming wetlands can develop. In such places, water levels remain high enough to slow the decay of plant material, allowing thick accumulations of partially decomposed leaves, stems and roots to build up over time. Conifer trees adapted to wet conditions together with tree ferns and other water-tolerant plants dominate the woody layer, while early flowering plants contribute to the ground layer and to the shrubby tier where conditions allow. In these settings, angiosperms often appear as small light-demanding elements in openings on raised hummocks or along microtopographic highs that experience brief periods of aerated soil between episodes of saturation. Their presence is recorded in the fossil record by leaves and seeds in the upper parts of peat sequences and by pollen in associated lake and floodplain deposits. On lake margins, flooding and drying cycles play a central role in structuring plant communities. During periods of higher lake-level waves and currents erode parts of the shore, transporting plant material into deeper water and burying some stands under beach or nearshore sediments. During lower stands, new strips of shoreline are exposed with moist but not inundated sediment, providing a substrate for rapid colonization. Early angiosperms that can complete their life cycle quickly are well positioned to exploit these newly exposed surfaces, establishing temporary populations that stabilize the sediment and add organic matter before the next phase of flooding or erosion. Fossil assemblages that include dense mats of small leaves and stems in nearshore lake deposits overlain by coarser sediments may represent such short-lived colonization events. Farther toward the coasts, where rivers meet inland seas or open oceans, lagoons and estuaries occupy sheltered embayments behind barrier complexes or within irregular shorelines. In these environments, water salinity varies with distance from river mouths and with tidal and storm conditions. Inner parts of lagoons close to freshwater inputs can be nearly fresh at some times, and brackish at others, while outer parts are more consistently influenced by marine water. Along the edges of these water bodies on low levees, tidal flats and back swamp areas, plant communities adjust to both salinity and water level fluctuations. Conifers tolerant of periodic flooding may occupy slightly elevated ground, their roots exploring saturated but not permanently submerged soils. In depressions and along tidal channels, herbaceous plants and shrubs that tolerate or require high moisture and occasional salinity establish dense cover. Evidence for vegetation in these zones comes from assemblages of plant fragments in lagoonal mudstones and fine sands where conifer twigs, fern fronds and leaves of flowering plants occur together with shells and other marine or brackish indicators. In such lagoon margins, early angiosperms appear as part of mixed, low vegetation, on point bars, levees, and abandoned small channels. They can occupy newly formed bars where fine sand and silt accumulate, as well as the tops of natural levees built by repeated overbank flows. Because these surfaces are frequently disturbed by lateral channel migration bank failure, and storm surges plants that can germinate and grow quickly have an advantage in establishing after each disturbance event. The fossil record of these settings sometimes shows thin plant-rich horizons between coarser channel deposits, reflecting short intervals when vegetation took hold on stabilizing substrates before being buried by later events. In deltas that protrude into lagoons or shallow seas, a similar pattern occurs. Distributary channels carry sediment onto the delta plain, where it is deposited as levees and crevassed splays or delivered directly into interdistributary bays. Between the channels, low-lying areas are occupied by wetlands with varying degrees of salinity and flooding frequency. In the upper parts of some early Cretaceous deltaic successions, pollen and macrofossil data together record increased representation of angiosperms in these interdistributary areas. Their leaves and seeds become more common relative to older groups in sediments, interpreted as overbank and back swamp deposits, while in the main channel, belts and nearshore marine zones, the plant debris remains dominated by conifers and other gymnosperms. This suggests that flowering plants find particular success in the quieter, more protected parts of the delta plain, where frequent but not catastrophic disturbance maintains open conditions and reliable moisture. In river-fed wetlands farther inland away from direct marine influence, seasonal changes in discharge produce cycles of flooding and drawdown that influence plant zonation. During high-flow periods, water spills out of channels and across floodplains covering low-lying areas and sometimes extending into adjacent swamp forests. Plants in the lowest positions must tolerate periods of submergence, while those on slightly higher ground experience only short-term inundation. When waters recede, exposed surfaces range from saturated mud to drying silt. Early angiosperms that germinate as floodwaters fall can establish on these freshly exposed surfaces, taking advantage of high nutrient availability from recently deposited sediment and organic matter. Ferns and horsetails with their spores and rhizomes also respond quickly, and the resulting plant cover is often a mixture of flowering and non-flowering taxa. The degree to which angiosperms dominate these temporary communities varies by region climate and the stage in their evolutionary expansion, and the fossil record captures a range of outcomes from almost purely fern-dominated carpets to mixed stands in which flowering plants are strongly represented. Lakes and wetlands also play a role in connecting plant communities across the landscape by acting as traps for transportable propagules. Seeds, fruits, and vegetative fragments of early angiosperms can be carried by streams into lakes and lagoons, where they either sink quickly near shore or float for some time before settling. In calm waters, some may remain viable, long enough to germinate on suitable substrates along the margins, thus extending the distribution of flowering plants from their original growth sites. Even when they do not establish their remains, accumulate in sediments providing records of nearby vegetation. The orientation and degree of abrasion of plant fragments in lake and lagoon deposits help paleobotanists distinguish between locally derived material and long-transported debris, though uncertainties remain especially where repeated reworking has occurred. In cooler or more seasonal climatic zones, lakes may experience stronger temperature differences between seasons, with possible ice cover in winter and mixing of water columns during spring and autumn. These changes in physical conditions influence nutrient cycling and productivity, which in turn affect plant growth along shorelines. Wooded swamp margins with conifers and ferns may give way to more open sedge or rush-dominated shores during parts of the year, with early angiosperms occupying niche positions where conditions are briefly favourable. Fossil evidence from high-latitude lake deposits that contain both gymnosperm and angiosperm leaves, together with spores and pollen adapted to cooler climates, supports the idea that flowering plants reach at least some temperate and subpolar wetland environments before the end of the early Cretaceous, though their diversity there is limited compared with lower latitude basins. Across all of these wet habitats, from inland lakes and peatlands to coastal lagoons and deltic back swamps, the presence of water alters the way disturbances act and the way plant communities recover. Flooding can uproot some plants, while leaving others largely unaffected depending on rooting depth and flexibility of stems. Sediment deposition can bury seedlings and low stems while providing new surfaces for future colonization. Fire is generally less common or more constrained in the wettest zones, but can sweep grass-like or shrubby belts on better-drained margins, resetting those communities. Through these cycles, flowering plants become integral components of many wetland and shallow water edge communities. They do not replace older groups outright, but they occupy important places in zone after zone, long water level gradients from the shallow margins of lakes and rivers to the slightly raised ground of levees and lagoon shores. In doing so, they contribute new types of leaves, stems and rooting patterns to environments where high moisture, fine sediments, and frequent physical change are the norm. The early Cretaceous wetlands, lakes, and lagoon margins are therefore key settings for the early diversification and spread of angiosperms, as well as for the continued success of conifers, ferns, and other long-established plants that share these water-influenced landscapes. In the early Cretaceous plant world, flowers are not yet the large complex structures familiar from many modern environments, but their essential parts are already present in a variety of small and relatively simple forms. A flower, in basic terms, is a short shoot that bears organs specialized for reproduction, including structures that produce pollen and structures that contain ovules, the precursors of seeds. In flowering plants, the ovules are enclosed by tissues that together form a carpel, and one or more carpels may be present in each flower. Around these reproductive organs there may also be leaf-like parts, sometimes organized as an outer set of protective segments, and an inner set that can function in attracting pollinators, though in the earliest known flowers the distinction between these sets is often subtle or absent. The fossil record of the early Cretaceous preserves partial and in some cases fairly complete examples of such structures in a range of sediments, mostly fine-grained rocks deposited in lakes, quiet river channels and low-energy floodplains, where delicate organs can be buried gently. Some of the best preserved early flowers are found as compressions or impressions, where the original plant tissues have been flattened between layers of sediment and later transformed into thin films of organic material or hollow molds. In these fossils, the three-dimensional form is compressed into two dimensions, but outlines and relative positions of parts can still be seen. Small, reproductive axes bearing clusters of what appear to be carpels and associated filaments are visible as patterns of curved lines and ovals, sometimes with enough detail to distinguish individual units. In a few cases, permineralized fossils in which mineral-rich water infiltrated plant tissues during burial and replaced or filled their cells preserve more detailed internal structures, allowing the shapes of ovules and the arrangement of tissues within carpels to be studied under microscopes. Even so, the record is incomplete, and many interpreted flowers rely on a combination of external form and position within the plant to infer function. Early Cretaceous flowers are often small, measured in millimeters to perhaps a few centimeters across. Some appear to be borne singly at the ends of short stalks, while others occur in clusters along axes that may represent inflorescences, that is, groups of flowers arranged on a common stem. In many fossils, the outer parts consist of a series of narrow leaf-like segments attached around the base of the flower. These segments, analogous to sepals and petals, can be uniform in size and shape or show modest differentiation. Their surfaces are usually smooth or only weakly ornamented in the preserved material, and their original textures and colours are unknown. Inside this outer ring, delicate filamentous structures interpreted as pollen-bearing organs or stamens radiate or stand upright. At their tips, swollen regions likely represent anthers, the parts that release pollen. Surrounding or central to these stamens are one or more carpels, sometimes free and sometimes fused into a single compound structure. The carpels may be elongated or rounded and contain ovules attached along their inner surfaces. In longitudinal or cross-sections of well-preserved specimens, the arrangement of ovules within carpels can be observed, revealing different patterns of attachment that later become important in distinguishing major angiosperm groups. The way these organs are assembled in early flowers suggests a range of basic morphologies. Some flowers appear elongated and streamlined with carpels arranged along a central axis, and few, if any, showy outer segments. Others are more compact, with a clearer distinction between an outer perianth, the collective term for sepals and petals, and an inner cluster of stamens and carpels. In several early Cretaceous species, the carpels are borne in an exposed manner, with little or no specialized structure forming a closed chamber around them, although their ovulars are still enclosed by carpal tissue. In others, carpels are fused or partially enclosed, forming an ovary, with a single or multiple chambers. Stigmatic surfaces, the regions where pollen would land and germinate, are sometimes indicated by roughened or expanded areas at the tips or along the margins of carpels, but in most cases the precise nature of these surfaces is not clearly preserved. The orientation of early flowers on the plant can be inferred when flowers remain attached to stems, or when associated leaves and axes are preserved in place. Some appear to be borne on slender flexible stems that suggest positioning away from the main axis of the plant, possibly in positions that would expose them to moving air or to the activity of animals. Others are more tightly integrated into leafy shoots. Aquatic or semi-aquatic early flowering plants sometimes show reproductive structures attached to stems that likely floated or lay near the water surface, with flowers arranged to access air above the water. Terrestrial forms may carry their flowers along upright shoots. However, in many fossil assemblages, flowers are detached from their parent plants and occur as isolated pieces, making it difficult to reconstruct their original position and orientation with certainty. The degree of symmetry in early flowers varies. Some show radial symmetry, in which parts are arranged as repeating units around a central axis, while others show hints of bilateral symmetry, with one plane dividing the flower into mirrored halves. Radially symmetrical flowers can accommodate interactions with potential pollinators approaching from multiple directions, while bilateral symmetry can relate more closely to specific approach routes. In the early Cretaceous, both patterns are present, though the details of how they relate to the behavior of any pollinating animal remain largely speculative, and the available fossils are not sufficient to firmly connect particular flowers to particular pollination systems. From a structural standpoint, many early angiosperm flowers display features consistent with wind or generalist animal pollination, such as relatively exposed anthers and stigmas and limited protection from the outer perianth. The internal anatomy of early flower parts, where preserved, reveals tissues consistent with water conduction support and reproductive function. Filaments of stamens contain vascular strands that supply nutrients and water to the developing anthers. Carpels contain vascular bundles that extend into ovules, supplying them during development. Cell wall patterns in some permineralized specimens suggest the presence of strengthening tissues that support the flower during development, and anthesis, the period in which flowers are open and functional. Yet many fine details of anatomy, such as those related to nectar production or specific mechanical devices for pollen release, are not clearly recorded in early Cretaceous material. The fruits that develop from these early flowers show multiple forms. In some cases, the carpel or group of carpels matures into a simple dry structure that splits open along defined lines to release seeds. In other cases, the enclosing tissue remains closed and either becomes thin and papery or thick and fleshy, though the degree of fleshiness is difficult to judge from compression fossils alone. The number and size of seeds per fruit vary, with some early angiosperm fruits containing one or a few relatively large seeds and others containing many small ones. The shapes of seeds and enclosing tissues as preserved suggest different potential roles in protection, dispersal, and dormancy, but the actual pathways by which these early fruits moved through their environments are addressed more fully by examining interactions with water, wind, and animals in other contexts. Within the early Cretaceous record, there is a clear increase through time in the variety of flower and fruit forms. Early in the period, only a few basic morphologies are present in any given region, but later strata show more diverse arrangements of perianth parts, stamens, and carpels. Some flowers develop more elaborate perianth structures with multiple whorls of segments and possible differentiation into distinct sepals and petals. Others refine the organization of stamens and carpels with more precise patterns of fusion, insertion, and number. Nonetheless, even the later early Cretaceous assemblages lack many of the extreme specializations seen in later epochs, and most flowers are still relatively small and simple. It is important to note what is not known about early Cretaceous flowers. Their colors are not preserved in most cases, though indirect inferences about pigmentation can sometimes be made from the chemistry of fossil tissues. Their scents, which in modern flowers play critical roles in attracting animal visitors, leave no direct trace in the rocks. The timing and sequence of developmental stages, including bud formation, opening, and senescence, cannot be directly observed. Patterns of temporal separation between pollen release and stigma receptivity, which today influence rates of self-fertilization and cross-fertilization, are similarly beyond direct reconstruction. Consequently, while the basic architecture of flowers and fruits can be described from fossils, many aspects of their functional biology remain uncertain and must be treated cautiously. Taken together, the early Cretaceous fossil record of flowers and fruits indicates that by this time angiosperms already possess the core structural components that define the group: enclosed ovules within carpel seeds, developing inside fruits, and reproductive organs arranged on specialized shoots that can include surrounding perianth parts. These structures are expressed in a range of small, generally simple forms that occupy various habitats, especially along water margins and in disturbed ground. Within the limits imposed by preservation, these early flowers represent working reproductive systems that operate within the broader plant communities of the time, adding new configurations of reproductive organs and enclosing tissues to the established palette of cones, spores, and exposed seeds that characterize the rest of the early Cretaceous plant world. Once fertilization has taken place inside the small flowers of early Cretaceous angiosperms, the visible signs of reproduction shift from pollen and ovules to fruit seeds and young shoots. These structures represent the stages by which new individuals are produced, protected, and established in the varied landscapes of lakeshores, floodplains, forests, and coastal plains. In the fossil record, fruits and seeds are preserved as compressions, impressions, casts, and in some cases permineralized specimens that retain internal anatomy. Together with the remains of seedlings and juvenile stems, they provide a window into how early flowering plants package their offspring, how those offspring are built, and how they begin life on the ground or in shallow water. In many early Cretaceous deposits, small dry fruits are especially common. These often appear as tiny elongated or rounded bodies a few millimeters to perhaps a centimeter in size, with surfaces that may be smooth, faintly ridged, or marked by subtle lines that reflect underlying tissue organization. Some represent single carpels that have matured into indehiscent fruits, meaning they do not split open when ripe, enclosing one seed or a small number of seeds that are released only when the fruit wall decays or is broken by external forces. Others correspond to carpels that split along one or more seams, with fossils preserving either the entire structure or one of the opened valves. The distinction between these basic fruit types can be made where detail is sufficient, but many fossils are isolated fragments whose exact mode of opening cannot be confidently reconstructed. Seeds enclosed within these early fruits vary in shape and proportion. Some are near spherical, others oval, flattened, or elongated. In permineralized examples, the seed coat or testa can be seen as a layered sheath around the embryo and stored food tissues. In some taxa, the outer layers of the seed coat are thickened and robust, suggesting a design that resists physical damage and possibly permits passage through abrasive environments such as coarse sediment or digestive systems. In others, the coat is thinner and more delicate, hinting at seeds that germinate relatively quickly after dispersal or that depend on burial in fine protective substrates. The size of the embryo relative to the volume of stored food, usually in the form of endosperm or a similar tissue, also varies. Some early angiosperm seeds contain relatively small embryos, implying that substantial development occurs after germination, while others hold larger embryos that may be more advanced at the time of dispersal. Fossil seeds can be found both within fruits and as isolated remains in sediment. Isolated seeds may have been transported away from their parent plants by water gravity or animals, or they may have been released when fruits decayed in place and later dispersed by erosion. In lacustrine and floodplain deposits, concentrations of small seeds often occur in fine laminae, or in particular bedding surfaces indicating episodes when seeds accumulated in low-energy settings, such as quiet backwaters, overbank ponds, or the resting stages of channel migration. These concentrations provide clues to the original abundance of seed-producing plants, and to the typical environments in which seeds came to rest. In some assemblages, seeds of angiosperms are mixed with those of conifers and other gymnosperms, as well as spores of ferns and other spore-bearing plants, reflecting the integration of angiosperms into existing reproductive landscapes. Early Cretaceous fruits show a range of attachment and enclosing configurations that can be related with caution to possible dispersal roles. Some small dry fruits bear winged-like extensions or flattened margins that would increase surface area relative to mass. In modern plants, similar structures promote movement in air or water by catching currents and slowing descent, and it is reasonable to suggest that early Cretaceous fruits with comparable forms could experience enhanced transport by wind or surface flow. Other fruits show hooks, spines, or surface irregularities that might catch on passing objects, although the specific interactions involved are not directly recorded and may have included both contact with plant litter and contact with animal bodies. There are also early examples of fruits with thicker, more substantial enclosing tissues that could be interpreted as proto-fleshy structures. In compression fossils, these are inferred from the thickness and texture of the preserved wall, from differences in preservation between the fruit wall and the seed, and from analogies with later angiosperms. However, the degree of true succulence or softness is difficult to gauge because fossilization tends to flatten and homogenize tissues. As a result, while it is plausible that some early Cretaceous fruits contained water-rich or nutrient-rich tissues that could attract animals, definitive evidence for such interactions is limited, and interpretations remain cautious. Seeds and fruits are not only transported as intact units. In some sediments, fragmented fruits and partially degraded seeds are found together with woody debris and leaf fragments, suggesting that mechanical breakdown during transport and after burial is common. This fragmentation complicates interpretation, but also reveals that many seeds and fruits are sufficiently robust to persist in transport, long enough to be incorporated into sedimentary deposits, rather than decaying rapidly at the surface. The resistance of seed coats to decay partly controls how likely it is that a given seed type will appear in the fossil record, so the seeds we see may represent a biased sample, favouring tougher structures. Nevertheless, taken in context with other plant remains, they provide a useful picture of the range of reproductive packaging strategies in early angiosperms. Once seeds germinate, the structure of young shoots and seedlings determines how successfully new individuals establish in the early Cretaceous environment. Direct fossils of seedlings are rarer than seeds and fruits, but there are examples of small plants preserved with simple root system, short stems, and a limited number of leaves that can reasonably be interpreted as juvenile angiosperms. In these fossils, the first leaves or cotyledons are sometimes visible attached near the base of the stem, with subsequent leaves showing more complex shapes and venation patterns. The venation of young leaves is an important characteristic because it influences how efficiently water and nutrients are transported within the leaf and how damage is localized or bypassed. Early angiosperm leaves often show reticulate venation, in which numerous small veins branch and reconnect to form a network. In juvenile leaves, these networks may be simple, but even simple networks provide redundant pathways for fluid movement and support. This contrasts with many contemporary gymnosperm leaves, which tend to have fewer, more parallel veins. As seedlings grow into small plants, their stems thicken and may develop woody tissues that provide mechanical support. Fossil stems of young angiosperm show vascular arrangements that include vessels specialized water conducting cells that differ from the trahides found in most gymnosperms. The presence of vessels allows for more efficient water transport in some conditions, especially in larger plants, though in small seedlings the advantage may be modest. Still, the combination of vessel-bearing stems and reticulate leaf venation allows young angiosperms to maintain water supply to leaves while exploiting changing microenvironments on floodplains, lake margins, and forest floors. Roots of young angiosperms, where preserved as casts or permineralized structures, typically show fine branching patterns that spread through upper soil layers. Such root systems can rapidly explore the near-surface region for moisture and nutrients, particularly in freshly deposited sediments, where competition from older roots is low. In contrast, many gymnosperm seedlings also produce fine roots, but the balance between shallow and deeper rooting may differ. In mixed communities, this difference in rooting pattern can allow angiosperm seedlings to occupy slightly different soil niches, taking up resources from the topmost horizons, while other plants draw on deeper reservoirs. When seedlings establish on disturbed surfaces such as freshly deposited silt after a flood or thin ash layers, after a volcanic event, early growth is influenced by the physical and chemical properties of the substrate. Fine-grained, nutrient-rich sediments can support rapid root expansion and shoot growth, especially when combined with adequate moisture and moderate temperatures. By contrast, coarse, nutrient-poor sands or gravels impose constraints on water retention and nutrient availability, and seedlings may experience high mortality unless they reach deeper layers quickly or find microhabitats where finer sediments and organic matter have accumulated. Fossil evidence for the success of seedlings in different settings comes primarily from the relative abundance of angiosperm remains in successive sedimentary layers and from the presence of root traces in paleosols. In some early Cretaceous successions, paleosols show dense networks of fine root traces consistent with repeated establishment of small plants, including angiosperms on surfaces that experienced intermittent stability between disturbance events. The spacing and penetration depth of these traces help to infer rooting behavior and indirectly the capacity of seedlings to survive short, dry spells or waterlogged conditions. In wetlands and lake margins, seedlings may face challenges related to water level fluctuations. Germination that begins during a period of low water can be interrupted if water levels rise, submerging young plants. Some early angiosperms likely tolerate temporary submergence, especially those adapted to aquatic or semi-aquatic habitats, but many terrestrial forms would be sensitive to prolonged flooding. Fossil assemblages that show repeated layers of small plant remains overlain by sediments deposited during higher water conditions illustrate this cycle of establishment and loss. In such environments, successful recruitment of new individuals may depend on synchrony between germination timing and the seasonal or episodic pattern of water level change. Young shoots also interact with the surrounding community structure. In dense conifer forests, seedlings must grow under reduced light, making shade tolerance or rapid vertical growth important traits. In more open wetlands and on disturbed floodplains, light is abundant, but competition for space and resources with ferns, horsetails, and other low vegetation can be intense. The relatively flexible growth habits of many early angiosperms, including the ability to produce multiple stems, branches, or leaves from low nodes, may have allowed them to adjust to different light conditions and to recover from partial damage. Fossil stems that show repeated branching near the base and leaves attached along short internodes support the presence of such growth forms. As seedlings develop into larger juveniles, their fruits and seeds begin to contribute to the local seed rain, reinforcing their presence in the habitat. The point at which an individual reaches reproductive maturity depends on growth rates, which are not directly measurable from fossils, but can be constrained by comparisons with growth ring patterns in wood and by the spacing of nodes and leaves on stems. Early angiosperms in disturbed resource-rich environments may reach reproductive size relatively quickly, enabling them to complete several generations within the time it takes a conifer seedling to reach similar size. This difference in generation time could influence how plant communities respond to repeated disturbances, but within the early Cretaceous, the available evidence supports only the broad conclusion that angiosperms are well represented among small, fast-cycling plants along water margins and on frequently renewed substrates. Overall, the early Cretaceous fossil record of fruit seeds and young shoots portrays flowering plants as lineages that already use enclosed seeds in diverse fruit packages, with seed coats ranging from fragile to robust, and with juveniles displaying vein networks, vessel-bearing stems, and fine roots suited to exploiting heterogeneous disturbance-dominated environments. These reproductive and developmental traits operate in parallel with and amid the established cones-naked seeds and spore-based strategies of other plants. Within the bounds of what can be observed, early angiosperms show a capacity to generate, protect, and establish new individuals efficiently in the physical settings available to them, contributing steadily to the reshaping of plant communities across floodplains, lake shores, wetlands, and mixed forests of the early Cretaceous world. In the early Cretaceous, flowers and other reproductive structures of angiosperms share forests, wetlands, and open floodplains with many insects and vertebrates that can move pollen, disturb flower parts as they feed, and occasionally use fruits and seeds as food. The strength and specificity of these interactions are difficult to reconstruct, but several lines of evidence show that by this time, flowers are already part of a landscape where air, water, and animals all contribute to pollen movement. The clearest direct evidence comes from insects preserved with pollen. In some early Cretaceous amber, tiny flies, thrips, and beetles are trapped together with scattered pollen grains. Under magnification, the grains adhere to hair scales and fine projections on insect bodies, especially on legs, mouthparts, and the underside of the thorax. In a number of specimens, the pollen belongs to gymnosperms, showing that cone-bearing plants continue to rely on both wind and animal visitors. In other inclusions, pollen grains have shapes and wall structures that match angiosperm pollen, from the same formations indicating that at least some insects visit flowering plants and carry their pollen even if their visits are not yet tied to highly specialized flower forms. Additional clues come from insects preserved in fine-grained sediments. Compression fossils of beetles, flies, and other small insects occur in the same layers as flowers and leaves, and in a few cases internal remains of pollen are preserved in mouth parts or gut contents. Some beetles have mandibles and grooves suited to feeding on pollen or soft tissues, while certain flies show proboscises capable of probing into small crevices. Thrips, which today often feed on pollen and plant juices, are already present with piercing mouthparts and bodies that can slip between sepals and petals. All of these forms have the basic anatomical tools to act as flower visitors and potential pollinators, though the fossil record rarely pairs a specific insect directly with a recognizable angiosperm flower. The structure of many early Cretaceous flowers agrees with the idea of generalist insect visitation. Flowers are typically small and relatively open with exposed stamens and carpels and modest outer parts. Some occur in clusters along short axes, bringing many reproductive structures together in a confined space. In modern settings, such arrangements favour small insects that walk across several flowers, collecting food resources and transferring pollen as they move. Early angiosperms with these features fit well within a pollination regime in which non-specialized beetles, flies, and thrips visit a range of plant species, including both flowering plants and gymnosperms, rather than engaging in tight one-to-one associations. Wind still plays a major role in pollen dispersal. Many conifers and other gymnosperms produce large quantities of light dry pollen that travels through the air, and their cones and pollen grains are abundant in early Cretaceous rocks. Some angiosperm flowers likely make use of wind as well, especially small, inconspicuous flowers with many stamens and simple perianths. Clear examples of fully developed wind pollination syndromes, such as long dangling stamens and strongly feathery stigmas, are better documented in later floras, but the coexistence of simple flowers and large volumes of airborne pollen makes mixed wind and insect pollination plausible. In wetlands and aquatic habitats, water can assist in pollen transfer over short distances. Plants with reproductive structures near the water surface may release pollen that drifts in thin films before contacting receptive surfaces on neighbouring plants. Direct fossil evidence for this mode is sparse, because pollen moved by water is not easily distinguished from pollen moved by air in sediments. However, the positioning of some early angiosperm reproductive organs at or just above water level suggests that water-mediated pollen movement is possible in these environments. Vertebrates are also present in early Cretaceous ecosystems and could interact with flowers, though direct evidence is limited. Small mammals, many of them insectivorous or omnivorous, move through understory vegetation and along low branches. As they forage, they may brush against flowers, picking up and depositing pollen on fur. Early birds and small feathered dinosaurs, some with beaks or teeth suited to picking small food items from foliage, also frequent vegetation where flowers occur. No fossil shows a mammal, bird, or non-avian dinosaur clearly engaged in pollination during the early Cretaceous, so any such roles must be treated as incidental. The simple open structure of most early flowers also suggests that if vertebrate contacts occur, they do so without elaborate morphological specialization. Tiny arthropods such as mites, recorded in amber and inferred from modern analogues, live on and around plant surfaces, including reproductive structures. They feed on pollen, fungal spores, and plant fluids, moving only short distances but often in great numbers. While each individual may transfer pollen only within a small area, their activity further ensures that pollen is not static on flowers, but is redistributed at the scale of inflorescences and nearby shoots. The microscopic structure of early Cretaceous pollen grains adds another layer of information. Some angiosperm pollen has sculptured outer walls with spines ridges or reticulate patterns that would improve adhesion to insect cuticle. Other grains are smoother and more uniform, more consistent with wind transport. When particular pollen types with adhesive features are abundant in sediments associated with angiosperm flowers, it is reasonable to infer that animals often carry them even if the specific pollinators remain unknown. At the same time, the presence of both sculptured and simple pollen in the same deposits points toward a mixture of pollination modes within the same plant communities. Taken together, these lines of evidence support a view of early Cretaceous pollination as a composite system. Angiosperms enter a world where insects are already diverse and familiar with feeding on pollen spores and plant tissues, where wind disperses large quantities of gymnosperm pollen, and where water moves fine particles through wetlands and shallow lakes. Early flowers are small and accessible, suited to visits by many types of insects, but not restricted to any one group. Highly specialized mutualisms, such as those seen later between particular flowers and specific bees, moths, or birds, are not clearly documented at this time. Instead, flowers' pollen and visiting animals interact in flexible ways with wind water, generalist insects, and perhaps incidental vertebrates, all contributing to pollen transfer. Within this broad, mixed regime, the basic reproductive structures of angiosperms function effectively, allowing seeds and fruits to be produced, and early flowering plant lineages to persist and expand in the varied habitats of the early Cretaceous landscape. As flowering plants become more common in early Cretaceous landscapes, their leaves, stems, seeds, and fruits enter into existing networks of herbivory that already involve dinosaurs, early birds, mammals, and many kinds of invertebrates. Evidence for these interactions comes from bite marks on leaves, damage to reproductive structures, plant material preserved in coprolites, and in rare cases gut contents within vertebrate skeletons. Together, these traces show that angiosperms are incorporated into the diets of a range of animals, although in the early Cretaceous they share this role with long-established plant groups, rather than replacing them. One of the clearest records of plant-herbivore interaction is the pattern of damage seen on fossil leaves. In beds that preserve diverse angiosperm leaf assemblages, many leaves display holes, edge-feeding skeletonization, galls, and mining traces. Some of these feeding types are associated with chewing mouthparts, likely from beetles, orthopterans, or larvae of various insects. Others, such as narrow tunnels within the leaf blade, point toward leaf miners that live inside the tissue. The distribution and frequency of these damage types vary between leaf species and through time. In some assemblages, angiosperm leaves show levels and kinds of damage comparable to those on gymnosperm leaves in the same beds, suggesting that insects do not strongly distinguish between plant groups at this stage. In other assemblages, particular angiosperm leaf types show concentrated damage of a specific kind, hinting at emerging host preferences, though the herbivores usually remain unknown. Galls, which are localized swellings induced by small organisms, appear on some early Cretaceous leaves and stems. Their presence indicates that some herbivores not only feed on plant tissues, but also manipulate plant development to create protective structures. The exact gall makers are rarely preserved, but groups that often form galls today are already present in the fauna, and both gymnosperms and angiosperms host galling organisms. At a larger scale, dinosaurs and other vertebrates contribute to herbivory on angiosperms, especially in mixed forests and floodplain environments, where these plants grow alongside conifers, cycads, and ferns. Coprolites from herbivorous or omnivorous dinosaurs sometimes contain small fragments of angiosperm tissues, including pieces of leaves and possibly seeds, together with abundant material from other plants. In some cases, microscopic examination of coprolites reveals angiosperm pollen grains and cuticle fragments dispersed among gymnosperm debris. These findings show that dinosaurs ingest flowering plant material, though they do not always allow distinction between deliberate feeding and incidental intake, while browsing on mixed vegetation. Gut contents preserved within the rib cages of a few dinosaur skeletons offer additional information. Where preserved plant material can be examined, it consists mainly of conifer needles, fern fronds, and other gymnosperm tissues, but in some specimens small quantities of angiosperm leaves or seeds are also present. The sample size is too small for firm generalization, but these records confirm that angiosperms form part of the available forage in at least some habitats. Smaller vertebrates, including mammals and early birds, also interact with angiosperms as herbivores or omnivores. Mammal jaws and teeth recovered from early Cretaceous sites display a range of shapes, from sharp cusps suited to insectivory, to broader surfaces that can process plant material, and micro-wear patterns sometimes suggest abrasion consistent with eating seeds or other hard plant parts. Direct association between particular mammals and angiosperm seeds is rare, but the co-occurrence of small hard seeds and mammals with appropriate dentition supports the possibility that some mammals consume flowering plant diaspores. Early birds, many of which still retain teeth, also show varied diets, and even when vertebrates primarily target other plants, they may incidentally clip angiosperm shoots or strip leaves as they move through mixed vegetation. Invertebrate herbivores, especially insects, are likely the most frequent consumers of early angiosperms. In addition to leaf damage, some fossils show boring traces in stems and wood that resemble those made by beetle larvae and other boring insects. Permineralized angiosperm wood from the later part of the early Cretaceous sometimes preserves galleries filled with frass, along with remains of the insects themselves. These traces indicate that as angiosperms begin to develop woody growth and contribute to the shrub and small tree layers, they become substrates for internal feeders. External feeders, such as grasshopper-like insects and caterpillar-like larvae, are inferred from patterns of margin feeding and surface scraping on leaves. Sucking insects leave different kinds of traces such as small punctures and subtle discoloration, and in the fossil record their presence is suggested by body fossils, mouthparts adapted for piercing and sucking, and in amber preserved interactions with plant surfaces. The impact of herbivory on angiosperm growth and community structure can be inferred from damage frequencies and from comparisons between heavily and lightly browsed leaves in the same assemblages. In some deposits, a large proportion of angiosperm leaves bear at least some damage, while in others damage is limited to a minority of specimens. This variation probably reflects differences in local herbivore, abundance plant defensive traits, and environmental conditions. Some angiosperm leaves show thickened margins, tougher midribs, or concentrations of small veins near common damage sites, features that may reduce the cost of tissue loss or channel damage away from critical transport pathways. Chemical defenses are not directly visible in fossils, but the diversity of herbivore feeding types suggests that early angiosperms present a range of nutritional and defensive profiles. Herbivory also generates inputs of partially processed plant material into soils and sediments. Chewed leaves, discarded fragments, and excreta contribute to litter layers, where they decompose and release nutrients. In coprolites, plant tissues are broken down and mixed with mineral material and microbial communities, forming concentrated packages of organic matter that, when deposited, can enrich local soils. Overall, the available evidence indicates that early Cretaceous angiosperms participate fully in herbivore-plant interaction networks that involve both invertebrates and vertebrates. Their tissues are eaten, mined, galled, and bored alongside those of conifers, cycads, ginkgophytes, and ferns. Insects use their leaves and stems as food, and habitat dinosaurs and other vertebrates ingest their foliage and seeds to varying degrees, and mammals and birds may exploit their reproductive structures as part of broader diets. These interactions do not yet revolve around highly specialized relationships, but they are sufficient to integrate flowering plants into the existing web of consumption and nutrient cycling that characterizes early Cretaceous terrestrial ecosystems. In early Cretaceous landscapes, the seeds and fruits of flowering plants do not stay where they are produced. Once released from the parent plant, they move across floodplains, down rivers into lakes, and lagoons, along coasts, down slopes, and through vegetation, on or inside animals. This movement or dispersal determines where new individuals can establish and how flowering plants spread through environments already occupied by conifers, cycads, ginkgophytes, ferns, and other seed plants. The main agents of dispersal are water, wind, gravity, and the bodies and activities of animals, and the fossil record preserves indirect traces of all of these processes. Water is a dominant dispersal medium in many early Cretaceous settings. On river plains, seasonal floods carry leaves, twigs, fruits, and seeds away from parent plants. Some diaspores fall directly into channels, while others are washed from the forest floor when rising water overtops banks. The energy of the flow sorts this material by size and density. Coarser, heavier items, including woody fragments and thick-walled fruits, tend to travel shorter distances before settling on bars and channel margins. lighter objects such as small seeds and thin leaf pieces can be transported farther and deposited in quieter zones, such as point bar interiors, backwater ponds, and distal floodplains. This sorting is recorded in fluvial sediments, where layers rich in small seeds and fine plant debris alternate with beds dominated by larger fragments. In some early Cretaceous deposits seed, rich laminae occur repeatedly in overbank and crevasse splay sequences, showing that waterborne transport and concentration of angiosperm diaspores occur frequently. In lakes, seeds and fruits can move both along shorelines and into deeper water. Material delivered by rivers or local runoff enters the lake at inlets, where currents and waves redistribute it along the margins. Some seeds sink quickly near the point of entry, especially if they are dense or lose buoyancy as they absorb water. Others float for longer periods carried by surface currents and wind-driven circulation before reaching shore or quiet bays. In core and outcrop records of lake deposits, this behaviour is reflected in the distribution of plant remains. Near-shore facies often show dense accumulations of seeds, fruits and small stems, while deeper water faces contain fewer diaspores, more dispersed among planktonic remains and fine mineral grains. Where seed types can be identified, some are characteristic of plants that likely grew around the lake, while others may have arrived from floodplains farther upstream, indicating that lakes function both as local dispersal zones and as sinks for propagules from broader catchments. Along coastal plains, rivers carry seeds and fruits into estuaries and lagoons, where tidal currents, waves and density contrast between fresh and saline water influence their paths. In brackish settings, some diaspores may float at the surface of less dense freshwater layers before mixing with underlying marine water. Others may be trapped in tidal channels and back swamps. Fossil assemblages from lagoonal mudstones that contain a mixture of freshwater and marine organism remains together with diverse plant debris attest to this complex movement and mixing. The presence of angiosperm seeds and fruits in such deposits shows that at least some early Cretaceous flowering plants have diaspores that tolerate short exposure to brackish conditions and can be transported into coastal basins. Wind dispersal is another important pathway, especially for small light seeds and for fruits equipped with wings or extended surfaces. Some early Cretaceous angiosperm fruits and seeds show flattened appendages or narrow membranous rims that would increase drag and slow their fall through air. In modern plants, similar features allow seeds to be carried away from the parent tree by breezes and gusts. In the fossil record, these winged seeds and fruits often occur in fine-grained sediments, interpreted as low-energy overbank or lacustrine deposits, where they may have settled out of the air directly, or been washed only short distances after landing. Their orientations on bedding planes, sometimes with consistent alignment, suggest that wind direction, at the time of deposition, influences how they come to rest. Pollen grains, although primarily involved in reproduction rather than seed dispersal, also travel widely by wind, and their widespread presence in sediments from many environments shows that air currents move plant microscopic propagules over regional distances. Gravity acts continuously on all diaspores. On slopes and in forests, ripe fruits and seeds drop to the ground beneath parent plants, or roll downslope into hollows and depressions. In steep terrain near mountain fronts, rockfalls and debris flows can entrain seeds and fruits from vegetation on upper slopes and carry them onto alluvial fans. In more gentle landscapes, gravity assists in moving diaspores down small inclines towards stream channels and low-lying wetlands. Fossil soils and fine overbank deposits that contain high concentrations of seeds beneath inferred canopy positions likely record local gravitational accumulation, while horizons with more mixed plant assemblages may reflect additional water or wind transport. Animals add further complexity to dispersal patterns. Invertebrates such as ants, beetles and other small arthropods can move seeds and fruits over short distances as they feed or carry pieces back to nests. Their activity is not directly visible in the fossil record of the early Cretaceous, but modern analogues and the presence of these groups in amber and sediments make small-scale animal-mediated movement plausible. More conspicuously, vertebrates can disperse seeds both externally and internally. External transport or epizoochory occurs when seeds or fruits adhere to feathers, fur or skin. Some early Cretaceous angiosperm fruits possess surface hooks, barbs or roughness that could catch on passing animals. At the same time, small dinosaurs, early birds with feathers and mammals with hair move through vegetation on forest floors along lake margins and across floodplains. While there is no direct fossil showing a seed attached to an animal from this period, the anatomical combination of adhesive fruit structures and mobile feathered or furred animals creates conditions in which external attachment is possible. Internal transport or endozoochory leaves more recognizable traces. Coprolites attributed to herbivorous or omnivorous dinosaurs occasionally contain identifiable seeds and fruit fragments. In some cases, these seeds show little physical damage, suggesting that they have passed through the digestive tract intact. If deposited in suitable habitats, such seeds could germinate at locations distant from their parent plants. Coprolites preserved in channel floodplain, and even coastal deposits indicate that animals deposit feces in a range of settings, providing opportunities for seed deposition in newly disturbed or nutrient-enriched microsites. For mammals and birds, direct evidence is limited, but their inferred diets and mobility suggest that they could also act as seed dispersers when consuming fruits or seeds as part of a broader diet. Dispersal by bodies of water, air, and animals operates together rather than in isolation. A single fruit might fall from a shrub to the forest floor, be carried a short distance downhill by rainwash encounter, a small stream, and move farther as part of a drift line of organic material, then finally come to rest on a point bar or floodplain surface. Along this path, it could be intercepted by animals broken apart or buried by sediment. In another case, a seed dispersed in a vehicle pellet could later be mobilized by water or incorporated into a soil horizon. These layered processes make it challenging to reconstruct exact dispersal routes from fossils, but they also ensure that diasporas have multiple chances to reach a variety of microsites. The patterns observed in early Cretaceous sediments suggest that despite these complexities, there are consistent tendencies. Water tends to move diaspores along drainage networks and into low points such as lakes, wetlands, and deltas. Wind spreads lighter seeds more widely across landscapes, with concentrations forming in sheltered zones and depositional basins. Animals contribute to shorter-range targeted deposition in habitats they use frequently, and to occasional long-distance movements when they travel between regions. The net effect is that angiosperm seeds and fruits, though produced in specific habitats such as floodplain forests, lake margins, or lagoon edges, can reach many other parts of the terrain. Within this framework, the dispersal strategies of early Cretaceous angiosperms appear varied but not yet extremely specialized. Some species produce numerous small, relatively simple seeds that rely mainly on air and water movement. Others invest in larger seeds with protective coats and enclosing tissues that may favor survival after passage through animal guts or in physically active environments. Some fruits show structures consistent with at least basic animal attachment or attraction, while others are streamlined and suited to fluid flow. All operate within a landscape where dispersal agents interact and where topography, sedimentation, and vegetation cover influence the final locations of seeds, the fossil record through distributions of diaspores in different faces, the presence of seeds in coprolites, and the forms of fruits and seeds themselves allows this combined picture to be drawn with reasonable confidence, even though many fine details of particular plant disperser relationships remain beyond direct observation. At the scale of tissues and cells, the early Cretaceous plant world gains further complexity, as flowering plants add new anatomical features to the existing range seen in conifers, cycads, ginkgophytes, and ferns. Fossil woodleaf cuticles and other permineralized remains from this interval preserve enough microscopic detail to show how water gases and nutrients move through plants, and how structural and chemical traits differ between groups. These inner characteristics influence how plants cope with the warm, often humid climates of the time, and how they occupy the diverse habitats of forests, wetlands, and river plains. In stems and trunks, the water-conducting tissue or xylem is a key focus. Most gymnosperms rely on tracheids, long narrow cells, with thickened walls and pits that allow water to pass between them. These cells provide both support and conduction. Early angiosperms, in contrast, begin to show xylem that includes vessels, which are series of shorter, wider cells stacked end to end, forming continuous tubes. In permineralized woods from the later part of the early Cretaceous vessel, elements appear alongside tracheids within the same stem. The vessel elements have perforated end walls where the cell wall is reduced or absent, allowing water to move more freely along the length of the vessel. Around them, fibers and parenchyma cells provide mechanical strength and storage. The presence of vessels suggests that some flowering plants can conduct water efficiently, especially in stems that must support leaves and branches above the ground. At the same time, the coexistence of tracheids and vessels shows that early angiosperm xylem is anatomically mixed and not yet uniform across all lineages or habitats. Growth rings in fossil wood record seasonal changes in growth rate. In some early Cretaceous woods, including those of both gymnosperms and angiosperms, rings with alternating bands of larger, thinner-walled cells and smaller, thicker-walled cells indicate that growth accelerates during favourable periods and slows during less favourable ones. In tropical or continuously wet climates, rings may be faint or absent, while in more seasonal environments they are clearer. This suggests that even within the greenhouse framework, plants experience variations in water availability, temperature, or light that imprint themselves on xylem anatomy. In some angiosperm woods, vessel size and density vary across growth rings, with larger vessels formed during times of greater water supply, and smaller ones during drier intervals. This pattern influences vulnerability to embolism, the formation of air bubbles that can block water flow, and points toward early adjustments in vessel design in response to environmental conditions. Outside the wood, the outer tissues of stems show protective and exchange features. Bark contains phloem, the tissue responsible for transporting sugars and other organic compounds from leaves to the rest of the plant. In many early Cretaceous specimens, phloem is not as well preserved as xylem, but the general organization with sieve elements and associated cells is inferred from structure in better preserved later fossils. The outermost tissues, including cork and periderm, protect against physical damage and water loss. In some permineralized angiosperm stems, resin canals or ducts occur similar in general concept to those in conifers, though often arranged differently. These ducts house resins or latex-like substances in life. Resins are complex mixtures of organic compounds that can deter herbivores and seal wounds. When they harden and are preserved, they form amber, which in turn traps insects and other small organisms. The presence of resin canals in early Cretaceous angiosperms indicates that chemical defenses and wound-sealing mechanisms are already part of their internal systems, operating alongside those of gymnosperms in mixed forests. Leaves provide another major window into microscopic anatomy. The outer surface of a leaf is covered by a cuticle, a thin layer of waxy material that limits water loss and protects against pathogens and physical damage. In fossil angiosperm leaves from the early Cretaceous, cuticles are often preserved as thin films that can be separated and examined. These cuticles show stomata, the pores through which leaves exchange gases with the atmosphere. Each stoma consists of a pair of guard cells that open and close the pore, sometimes accompanied by subsidiary cells. The density and arrangement of stomata on the leaf surface vary between species and between different parts of the leaf. In some early angiosperms, stomata are scattered evenly, while in others they cluster along particular zones. Stomatal density is influenced by atmospheric carbon dioxide concentration and by the plant's water relations. In high carbon dioxide conditions, plants can maintain lower stomatal densities and still achieve sufficient gas exchange, which also helps conserve water. Comparisons of stomatal characteristics between early Cretaceous angiosperms and coexisting gymnosperms combined with geochemical proxies support the view that atmospheric carbon dioxide is higher than present during much of this interval. Within the leaf interior, the arrangement of tissues guides light capture and gas diffusion. Many early angiosperm leaves show an upper layer of elongated cells, often referred to as palisade tissue, and a lower layer of more irregular, loosely arranged cells, often called spongy tissue. The palisade layer, located beneath the upper epidermis, is well suited to absorbing light, while the spongy layer with its larger air spaces facilitates gas movement. A network of veins composed of xylem and phloem runs through this mesophyll. Primary veins branch into secondary and higher order veins, forming a reticulate pattern that supports the leaf and distributes water and nutrients. This network also provides redundancy. If one vein is damaged by herbivory or mechanical stress, neighbouring veins can maintain supply to the affected region. In gymnosperm leaves of the same time, vascular arrangement is often simpler, with one or a few main bundles and less branching. The more complex venation of angiosperms at the microscopic level allows fine-scale control of water distribution and may support higher rates of photosynthesis under some conditions. In roots, microscopic anatomy reveals adaptations to different soil conditions. Root tissues include an outer epidermis, a cortex, and a central stele containing xylem and phloem. Root hairs' extensions of epidermal cells increase absorptive surface area. In permineralized roots from early Cretaceous sediments, fine branching patterns and abundant hair bases show that both angiosperms and other plants exploit the upper soil layers intensively. Within the cortex, mycorrhizal associations appear inferred from the presence of fungal structures embedded in root tissues. These symbioses, in which fungi assist with nutrient uptake in exchange for carbon, are widespread among modern plants and likely present in early Cretaceous angiosperms as well. Such associations influence plant nutrition, especially for phosphorus and nitrogen, and affect how plants respond to differences in soil chemistry across floodplains, uplands, and volcanic substrates. Plant chemistry leaves fewer direct macroscopic traces, but certain features can be inferred. The presence of resin canals in wood and bark, together with the production of amber, indicates synthesis of complex terpenoid compounds. Leaf cuticles incorporate waxes and other long-chain molecules that resist decay, contributing to the persistence of cuticle fragments in sediments. Some early Cretaceous leaves show darkened carbon-rich residues along their margins, and in veins suggesting concentrations of lignin, the structural polymer that stiffens cell walls and makes them more resistant to breakdown. Lignin content affects how quickly litter decomposes and how much carbon is stored in soils and sediments. Differences in lignin and other compounds between angiosperms and gymnosperms influence the composition of organic layers, even when these differences cannot be fully resolved at the molecular level in fossils. The chemistry of cell walls and internal tissues also affects how plants interact with herbivores and pathogens. Secondary metabolites such as phenolic compounds, alkaloids, and various defensive molecules do not preserve well in detail, but their former presence is suggested by patterns of selective herbivory and by the association of certain damage types with specific leaf taxa. Some leaves show restricted feeding only along softer tissues, while tougher, possibly more chemically defended regions remain intact. Others show characteristic patterns of avoidance around veins or margins. These patterns, together with modern analogues, imply that early angiosperms synthesize chemical compounds that influence feeding behavior just as gymnosperms do. Finally, the overall composition of plant tissues, including carbon to nitrogen ratios and the presence of more resistant compounds, shapes how plant material enters the sedimentary record. Woody tissues rich in lignin and resistant carbohydrates persist as larger fragments, while more labile tissues break down into finer organic matter. In early Cretaceous settings, the mixture of angiosperm and gymnosperm tissues, each with their own microscopic structures and chemistries, contributes to varied organic signatures in soils, peats, and sedimentary rocks. These signatures reflect the inner workings of plants and set the stage for the microscopic remains, such as pollen spores, and fine organic particles that form another important archive of early Cretaceous ecosystems. In the early Cretaceous, many of the most durable traces of plant life are not whole trunks or intact leaves, but microscopic grains, fine fragments, and thin organic films locked into layers of sediment that later become rock. Among these traces, pollen and spores are especially important. Pollen grains carry the male gametes of seed plants, including both gymnosperms and angiosperms, while spores serve as reproductive units for ferns, horsetails, and other spore-bearing plants. Both are produced in very large numbers and are small enough to be transported by wind and water over considerable distances. Once they settle onto soil water surfaces or directly into accumulating sediment, they can become buried and preserved. In rocks of early Cretaceous age, pollen and spores form dense assemblages that record in a time-averaged way the composition of plant communities across landscapes. Because these microfossils resist decay better than most soft tissues, they provide one of the most continuous archives for tracking the rise and spread of flowering plants. Under the microscope, early Cretaceous pollen and spores show a wide variety of shapes, sizes, and wall structures. Gymnosperm pollen grains are often relatively large and simple, sometimes with air sacs that assist in wine transport. Fern spores display characteristic surface patterns and aperture types. Angiosperm pollen, when it appears, is distinguished by features such as particular numbers and arrangements of apertures and sculpturing of the outer wall. As time progresses through, the early Cretaceous strata in many basins show a gradual increase in the diversity and abundance of pollen types attributed to angiosperms. In some sequences, the first occurrence of clearly angiospermous pollen is followed by an expansion in both the number of distinct forms and their proportion relative to gymnosperm pollen. This pattern does not appear everywhere at the same time, but where it occurs it provides a sensitive signal of angiosperm diversification, even when macrofossils are sparse. Spores and pollen do not remain where they are produced. Wind can carry them from upland forests into lowland basins and from dry interiors to coastal plains. Rivers wash them from soils into channels and transport them downstream where they settle with fine sediment in floodplains, lakes and shallow seas. As a result, a sample of pollen and spores from a single rock layer usually reflects a mixture of local and regional vegetation. Palynologists interpret such assemblages by combining knowledge of sediment transport with information about the plants that produce each type of grain. High proportions of spores, for example, may point toward extensive fern-rich habitats, while increases in certain pollen types can signal expansion of particular seed plant groups. In the early Cretaceous, the appearance of angiosperm pollen in assemblages dominated by gymnosperm pollen and fern spores indicates that flowering plants have joined existing plant communities rather than replacing them. The sediments that host these microfossils also preserve other organic signals. Fine plant fragments, cuticle pieces and amorphous organic matter accumulate in muds and silts, on floodplains in lakes, and on continental shelves. In some settings, especially under low oxygen conditions, this material builds up faster than it decomposes, forming organic rich layers. When buried, such layers function as reservoirs of information about productivity and preservation. The ratio of organic carbon to mineral content, the types of plant tissues present, and the degree of degradation all provide clues about whether sediments formed in well-oxygenated rivers, stagnant lakes, or restricted marine basins. In some regions, the early Cretaceous rock record includes dark organic rich shales, interbedded with lighter-coloured sandstones and siltstones. The shales often represent intervals when water columns in lakes or inland seas were stratified with low oxygen at depth suppressing decomposition. During such intervals, pollen spores and fine plant debris falling to the bottom are preserved with minimal alteration. The surrounding sandstones and siltstones reflect more energetic conditions such as river channel activity or storm reworking, during which coarse material accumulated and some organic matter was oxidized or fragmented. By comparing the contents of these rock types, scientists infer how environmental conditions fluctuated over time. Marine sediments add further context. On continental shelves and in shallow epicontinental seas, the settling of terrestrial pollen and spores from the atmosphere and from river plumes creates mixed assemblages that include both land-derived microfossils and remains of marine organisms. In early Cretaceous sections of this kind, changes in the relative abundance of terrestrial pollen and spores can reflect shifts in vegetation on adjacent continents, or changes in erosion and river discharge. The first widespread appearances of angiosperm pollen in these marine records show when flowering plants have become abundant enough on land for their microscopic products to be carried far offshore. Beyond microfossils and organic residues, the arrangement of rock layers themselves provides information about early Cretaceous ecosystems. Stacked sequences of channel sands, floodplain muds, paleosols and coal beds record repeated cycles of river migration, floodplain stability, and wetland development. Paleosols, or ancient soils preserved within these sequences, contain root traces, soil horizons and geochemical signatures that indicate the types of vegetation they supported and the climate under which they formed. Dense networks of fine root traces near the top of a paleosol suggest a cover of herbs and small shrubs, while larger, more widely spaced roots indicate trees. The presence of specific kinds of carbonate nodules, such as iron, accumulations or clay minerals, helps constrain rainfall patterns and seasonality. When angiosperm roots are present alongside those of other plants, they show that flowering plants have become parts of the communities that shape and are shaped by soil formation processes. Coal beds formed from accumulated peat in ancient wetlands are another archive. Their botanical composition, as revealed by microscopic analysis, shows which plant groups dominated the peat-forming communities at particular times. Early Cretaceous coals are often rich in conifer and fern material, but in some regions the proportion of angiosperm fragments increases toward the end of the interval, marking their growing role in swamp and mire vegetation. Taken together, pollen spores, organic residues, paleosols, coals, and the geometry of rock bodies form an archive of early Cretaceous ecosystems. Microfossils track broad patterns of plant presence and abundance. Organic rich shales and coals record productivity and preservation under particular redox conditions. Paleosols reveal details of local vegetation and climate. Stratigraphic stacking patterns document how landscapes responded to changes in sea level tectonics and sediment supply. Within this archive, the rise of flowering plants is visible in the prominence of angiosperm pollen, in microfossil assemblages, and in the presence of their tissues and roots within soils and wetlands of the early Cretaceous.